to do whatever you need to do to protect yourself. And if you would like masks or COVID tests, they are available at the registration desk. This is the Wi-Fi. It's also in the center of every table, so don't ask us. It's there. Attendee mobile app. This is great for allowing virtual and in-person attendees to all participate equally. You can ask your questions on the app using the GA4GH hashtag. So download or, wow, that's Slido, sorry. Attendee mobile app is for people in person to have access to all the agenda, the bios, and all of that. Code of ethics and community conduct. Please respect each other, be kind. Check out the Code of Ethics and Community Conduct on our website, and if you do witness or experience anything that you want to report, you can contact conduct@ga4gh.org or any member of the SAFE GA4GH committee, which is listed on the website. This is Slido, which allows everyone to participate equally. Navigate to sli.do in your web browser, or you can download the app and then search for GA4GH. You can ask questions throughout the talk, so you don't need to wait until the end. And then we'll pull up the questions at the end of each talk and go through them. And now I think I'm turning it over to David.
Good morning, everyone. I have the honor and privilege of introducing and emceeing this morning's sessions, and we're starting with a talk that I'm excited to hear Karen talk to us about her recent work on pangenome. Karen Miga, Dr. Miga, is an assistant professor in the biomolecular engineering department at UCSC. How many people know the mascot of UCSC? Not enough hands. You get to do the reveal. You get to tell them when you come up here. And an associate director of the UCSC Genomics Institute in 2019, she co-founded the Telomere to Telomere Consortium, an open community-based effort to generate the first complete assembly of a human genome. I thought we did that 20 years ago. No. Yeah. Additionally, Dr. Miga is the director of the Reference Production Center for the Human Pangenome Reference Consortium. Central to Dr. Miga's research program is the emphasis on satellite DNA biology and the use of long-read and new genome technologies to construct high-quality genetic and epigenetic maps of human pan and para and centromeric regions. You're going to hear a lot more about that in a minute. Karen, take it away.
Thank you so much. We are the mighty banana slugs, no known predators, for those of you who haven't seen Pulp Fiction, where it's kind of part of the story. So once again, I'm Karen Miga. I'm delighted to be here. This is absolutely the community that we need to speak with, work closely with, and I'm actually proud to say we have had a lot of wonderful efforts that have been intertwined with GA4GH since the beginning of our efforts, and I'm really proud to be standing here as part of the driver project announcement so we can make this even more fruitful as we move forward. So as we all know, as it was introduced, there's a need to modernize the human reference genome. We all know that it's foundational for biomedical research and clinical genomics, but like most technology, like your cell phone or your computers, after a couple of years, it's time for an update. And this is the same thing. We're talking about a genome that was constructed over 20 years ago. It's looking at a largely linear haplotype of largely one individual. And as we all know, since we study variation in this group, that it instantly extends some biases and also excludes sequence variation. And I'm very excited to stand here because it is a very exciting space right now to be in long read and assembly. And we are now seeing such gains in this space that it has allowed for us to have high production now of these near T2T or high quality genomes that we've never seen before. So many of you who may have been paying attention to a lot of the celebration back in April of 2022, we now have the first complete human genome. This was a celebration from the T2T consortium, which I'm showing here at the bottom, where we finally met all together last year face-to-face in Santa Cruz. And I'm showing the science cover because I want your eyes to see those bright red blocks. These are all the extensive gaps that were found on every single human chromosome, some of them extending tens of megabases in length, that were finally filled in. And we know in total what we're talking about is an introduction of about 200 million bases, and that this was finally resolved by a combination of long-read sequencing, really crediting 2 companies. This is PacBio, or the circular consensus sequencing, or HiFi, and Nanopore, with the advantage of having these ultra-long, or reads that can go for 100 kb plus. And what this has allowed us to see for the first time is new genome biology, new functional elements. We can talk about variants, but now we can talk about them comprehensively, end to end on each chromosome, and we can begin to expand catalogs of what's functional in a genome. And as I mentioned before, we're seeing this really rapid technology gain in this space, to where it's not just one genome anymore. We can start thinking about how we can knock down that technological barrier and start to reach complete reference assemblies of thousands of human haplotypes now. So although I took the stage as a co-founder of the Telomere to Telomere Consortium, I think if you're in the virtual water coolers of the HPRC, you would know that the T2T is part of the HPRC Assembly Working Group. We work side by side, and we're kind of teamed up now to try to create a better reference genome that has this high mark of quality and being comprehensive. Now, as you might have seen as well, there's been a lot of press about trying to move into this new space away from the prior human genome. This is a quote that was taken from The Guardian, where you have this healthcare standard genome, or the GA4GH 38, is mostly based on one American. And as we enter into an era of precision medicine, this bias clearly has drawbacks for much of the world's population. And this is a slide I see at almost every conference that I attend, so really crediting Martin et al. Back in 2019. But we know that we have a problem in genomics in terms of diversity, where most of our data sets here, I'm just showing GWAS, but this is clearly across the board, has an overrepresentation of individuals who are identify as European. So why is one genome not enough? Not to really belabor this point, but we know that there are changes or variants in our genomes that are large. These are structural variants, and they're difficult to identify with our current methods and our current reference genomes. We know that there are tens of megabases of these common polymorphic sequences missing from any individual reference, any individual in this room. And there's no single reference assembly, even the touted CHM13 T2T assembly, that can really be optimal for all people. It creates a reference bias. If you were to take short read sequences in a kind of a controlled benchmark data set and just run through your standard mapping and variant calling protocols right now, what you'd be missing is about 70% of these bigger events. And these bigger events are important. They actually can map to several regions that we know present medically important regions in our genome, such as HLA typing. Forgive the bottom of the slide, I don't know why that's popped up, but I really clearly want to emphasize HLA, apparently. But we know that that's important for transplants as well as autoimmunity, allergies, and drug reactions. The SMA locus, as well, is one of these leading causes of early infant death, 1 in 50 are carriers, and this is a medically important region. However, it's a complex region that contains multiple paralogs and is difficult to assess with short reads alone. And also when we start thinking about the CYP2D6, I said mouthful, it's really responsible for about 25% of our clinically used drugs, and variations in this gene can greatly affect how we interpret, how we can metabolize these drugs. This is just really pushing into this point that sometimes it's not the gene, but the components of the genes alone. Here I'm looking at the LPA gene, or LPA, which we know is associated with an increased risk of heart disease. And it contains these Kringle4 type 2 elements, as I'm trying to show you as the tiny little ticks. And based on how many you have a different, likelihood of having heart disease, and that having that kind of information about structural variation in your genome or in patient's genome could be very important for medically relevant loci. And of course, as introduced, I love satellite DNA, and I love these highly repetitive regions, and I think it would also be important to take this soapbox and say there's a lot in our genome we don't know about that could be medically relevant. Centromeres that we know are critically important for how chromosomes segregate. If you have an error there, it could lead to aneuploidies or chromosome missegregation, are clearly different between different individuals. There's a tremendous amount of variation we haven't yet started to sample. We know that gene families, that some are involved in the segmental duplications, can have copy number variation depending on the individual that you're studying. It could have a 1 to 6, as shown in this example for SMN. And this could have an influence as well as how we understand genome biology and organization. And now we're starting to think on the context of a whole chromosome. And there's been some really beautiful early studies that have shown for the acrocentric chromosomes, there are regions that are similar between the different acrocentrics that can lead to whole arm swapping. And understanding these big genomic features all the way down to the repetitive structural variants are things that we haven't even started to catalog yet that could lead to new insights into genome medicine. So let's talk a little bit about pangenome. We've been talking about a reference genome so far, but what I want to lean you into now is thinking about all of these high-quality T-to-T assembly references now. You have them organized in a multiple alignment. Imagine greater than about 1,000 of these. And if you could organize that now into something called looks like a string graph or a sequence graph, you can begin to see the shared regions and the regions that are different that can now present variation. And this is really the context of where we're taking this. It's a multiple alignment of near complete, if not complete, T-to-T chromosomes. And by having that type of reference base, you can begin to readily identify differences in variation using this type of graph. There's some key features of this graph I thought it'd be useful to really bring into this conversation. One is that it's important that it's open access. We make sure that the use of the assemblies, the sequence data, the technology, protocol, the standards, all of that needs to be something that anyone around the world could open their laptop and pull it in their clinic or at their research bench. And this is a special feature that we really don't emphasize enough for the HPRC, but I think it's important to really stress this here. For the initial Human Genome Project, there was no cell line to match. There was no donor match cell line. Even for CHM13, it's a difficult cell line to obtain and to grow. Here, all of these different references that we're working with have access to a biobanked LCL cell line, and in some subset, we're creating a panel of human iPSCs. And so this is really helping to also help promote studies utilizing these biorepository materials as well. Here's the part I'm hoping that everyone gets excited about in this room, because where we're wanting to move, of course, is more advanced pangenomic tool ecosystems and standards. We're already starting this, of course, with trying to build our workflows for reaching T-to-T assembly and production, QC, and also leading genome annotation and gene annotation. We need to think about how we're going to do read and assembly alignment to this graph and begin to go through variant calling. And there's lots of applications here that you could imagine could be useful for folks who are interested in precision medicine. One I'm listing on the bottom is just to advance CRISPR design guides and things of that nature utilizing this type of new structure. We are sharing all of our workflows right now and various sites. I'm trying to list them here for anyone who wants to go afterwards. And we've held a few workshops where we've been trying to teach folks how to actually implement some of these variant calling using our earliest pangenome, and those are available on YouTube. Oh, forgive me, if I can go back. And there's also an important point here that sometimes is missed. On the first slide, I emphasized how important it is to be open access of the data sets and how important it is to download this wherever you are. But there are a number of groups around the world, may they be medical or may they be groups who want to have data sovereignty, who will construct their own pangenome reference resource using pangenomic tools, but they may not share that same open access model. However, like the celebration of 1,000 genomes, where it was not only the release of the sequence material, it's also the tools, the formatting, the standards. I think the pangenome is gonna hold that torch as well. And by creating these types of tools, we are creating an ecosystem that people outside of the pangenome can begin to utilize that and build their own databases for their own designed research questions. And then finally, the human genome would not be as useful to folks if we just passed it out and it was just chromosome 1 and gave you a series of coordinates. These coordinates all have meaning. I like to put this here for when I talk to students about the human genome. We can think about it like Google Maps. If we just had a bunch of coordinate spaces, no one would use Google Maps. We want to know annotation. We want to know how to get from point A to point B. We want to look at various restaurants. And that is exactly what this needs to become as well. The human pangenome reference needs to be a one-stop shop for functional annotation, where if you were to go into this ecosystem, you're not only able to map and detect variants, but you can begin to get some information about those variants, how they're related to disease or how they're related to function. And so now I get to switch gears and I get to introduce you to the Human Pangenome Reference Consortium. This was really a call to action from NIH to improve the representation of the human reference genome, and by doing so, increasing global genomic diversity. The goal for our first phase was really to include at least 350 diverse diploid genomes and trying to represent common allele frequencies or global common allele frequencies of at least 1%. In doing so, we organized this an amazing team, of which I'm always so proud to represent on stage, of researchers with a ton of experience with long-read sequencing technologies, assembly, and I'm showcasing these at the bottom. You can see the T2T, VGP, as well as the HGSBC, where we have key leadership as part of our team. And although I don't have as many fun icons at the bottom, I really want to emphasize as well that we've organized an amazing group of thought leaders around pangenomic data structures and thinking about new tools, and this is another space where our team really excels. With all big science, this has to be highly collaborative, and what I'm wanting to show in this slide is kind of a clear understanding of how our organization is structured. You can imagine that this starts off with kind of population sampling and representation. We need to think deeply about technology and production, going into these assemblies, organizing those assemblies, as we already introduced, into this multiple alignment or this new data structure, and then how can we begin to fold this into the more international standards and outreach that's happening in this community as well. Hopefully, all of that, your eyes did not miss the most important part on the bottom, or an important contributor at the bottom, which is throughout every single step, we have an embedded ethics and policy team that really helps to contribute and strengthen our work. So if we were to start thinking about population sampling and representation, as I mentioned before, our goal is to comprehensively capture common variants, here described as a global frequency of 1%. There are clear challenges here. There are lots of unknown. For sure, we have an incomplete understanding of the full spectrum of human genetic diversity. We don't have that represented in any one database or even collectively. So understanding when we make that 1% mark is a bit of an aspirational goal. And there's a lack of consensus right now on the best approach for sample selection, so I'll just walk you through what we're doing, and we're taking multiple orthogonal methods. In the first phase, we're focused exclusively on the use of the 1,000 genome cell lines. These have the advantage of a compatible consent for open access use, and they also were able to grow up and serve as this production substrate for our long-read protocols. When we think about population sampling and representation, our teams have brought in 3 distinct models which we're operating around. The first one is really thinking about maximizing common variant diversity. Here we're leveraging existing sequencing data to iteratively sample to maximize common variant coverage across an out-of-sample data set. So if you have 1,000 genomes VCF, you could easily imagine how you could take a greedy algorithm or maximum var algorithm to begin to identify those individuals that maximally cover common variants in that set. In model 2, we're maximizing our genetic divergence. This is really taking into a model where we understand individuals have admixture and looking at clines through PCA and trying to develop methods that way. And of course, we understand as a consortium that there will be underrepresented populations that we need to actually go through and do due diligence with community outreach and support in order to have designated participants from that country join our group. And just to give you a feel for how we're performing so far, we took a kind of 4-year review, and we're looking at 1,000 genomes here in our evaluation method. And what we're finding is when we evaluate the sets that are currently in the pangenome production queue, we are currently covering 99.1% of these common SNVs that are available to us through 1,000 genomes. No one in our group would say that we're pleased with sticking with 1,000 genomes as kind of our sampling source. I think that what we're thinking about now is how can we begin to increase the breadth of outreach internationally, but our first step forward is through partnerships with the Biobank and the Streets of New York. This is really important because in partnership with Emer Kenny, shown here on the slide as our key PI, we have access now to over 70,000 patients that represent 160 countries and sequencing data that are available to our team for more than 40,000. We're also actively reaching out to other biobanks, such as the WashU Lewis Recruitment Center that's already been useful, and other consortiums for doing recruitment efforts with African American communities. This is, of course, big science at its best. I love to really emphasize the amazing skill of our various production centers. This is another place where I spend a lot of my headspace and time. We have a number of centers around the US where we're generating high-quality hi-fi, ultra-long data, as well as any kind of short-read data that's important for phasing, if it's trio-based sequencing with Illumina, or if it's a high-C-based data product like Cantata OmniC. And I always like to put on the slide, although sometimes I speed over it, this cluster of icons at the bottom. It's been incredibly useful for our team. This is one of our few working groups where we actually do have a lot of company partnerships, and this has really helped our team to be on the forefront, I think, of a lot of these new sequencing technologies. In particular, when we start thinking about long reads, our team had early access. We've been working directly with company partnerships, trying to onboard Revio, as well as performing the new duplex or the high-quality sequences that are available through ONT. And as soon as we get these data, we try to make them available to the community. So we also try to be a source for community innovation as well. Data management, this is something that we have been moving towards since the first phase of our grant and trying to identify how we can take all of these different data from all of the different centers, pull them into AWS where we have a Human Pangenomics bucket for our sequence data and assemblies, but ultimately we sync this all into Anvil and make sure that it's available on public biorepositories. And as I mentioned before, we do have various sites where all of the coding is available to researchers to make sure it's reproducible. We have organized an amazing team of long-read assembly methods experts. I will say I had a really heavy hand in developing our first Genome Center grant, and I've seen this pricing and assembly protocol for the last 4 years, and it has been an amazing triumph of how far we've come over a very short time. And where our team is moving now is into this more advanced automated diploid T2T assembly stage. This is really crediting 2 teams, Adam Philippi from NIH, as well as Hung Lee, who's from the Dana-Farber Cancer Institute, where we're now taking both the hi-fi and these very long ONT reads, and we're mixing them together. If I can, I'll walk you through a little bit more than that, to start to think about how we can create these long haplotype phase T2T assemblies. This type of partnership is critically important. This is one of the triumphs of working with these teams is Adam Philippi's due diligence to always chase perfection. And this is, the T2T consortium is now leading the charge to try to reach a QV100, or the most highly accurate reference benchmark known to the history of all genome assemblies. And this is wonderful. And what I'm trying to show you here is for HG002, where the red and blue are really distinguishing the maternal versus paternal haplotypes, because it creates a benchmark for our team as well to try to optimize our assembly strategies to really try to dial in that level of quality for production. And why is all this important? I've spent a lot of your time talking about assembly, but I really want to go back to the clinical components. And that is when we start thinking about complex sites, as I introduced earlier in my slide, like SMN1 and its paralog SMN2, we know that these reside on very complex genomic regions on 5q. And unequal crossing over can lead to variable copy numbers in these regions, and this could be very important for clinicians to understand some really horrible diseases like SMA, which we know is one of the leading causes, once again, of early infant death. And before, when we had these early assembly-based methods, what we were finding is if you were to open up that assembly graph, we were finding breaks, and it was an imperfect representation of this site. However, with our newer assemblies, we absolutely can make it clear across these very important medically important regions to represent. And we can even do this now across pedigrees. This is just giving you an example, since I mentioned that 1 in 50 are carriers, as we can begin to move this into looking at how its inheritance patterns happen between parents, grandparents, all the way to the provan. Now, we are celebrating the first release of a Human Pangenome. This is something that we had an alignment of 94 of these highly phased haplotypes. I'm trying to show you here, once again, that this was a celebration just recently of May of 2023. And here what we're trying to do is really illustrate kind of a blown up view of how this type of multiple alignment works, where you can see in blue versus orange the alignments, and then on the bottom that's represented as a graph. The input here, of course, is the FASTA sequences. These are the alignments, may it be HG38, as well as the T2T assembly and contigs from 44 of these diploid HPRC assemblies. The output is a GFA, or a graph format. It represents the multiple alignment of the input contigs, and each contig has a path in the graph. And we now have standard text-based formats that's supported by VG and other GFA tools. Now you can think graphs sound scary, but you can instantly, I want to use the next 2 slides to really kind of push for why this should not be as scary as some of the audience might be feeling, because there are real clear opportunities for more immediate adoption, and that's based on the shared coordinate space with HG38. For example, we have tooling in-house, really crediting Benedict Payton's team with VG Giraffe, to really look at short read sequence map to a pangenome, as we're trying to illustrate here with the little pink boxes over this sequence graph. However, the HG38 coordinates are on the bottom, illustrated in green, and so you can take these mapping and you can begin to look at the aligned reads projected to HG38, and then using Google Deep Variant Callers, you can begin to make your variant calls relative to those coordinates. And I've heard this from the first day, that all of the tools, I know this is not specific to just a pangenome, for adoption need to be dead simple to use. And so I would love for you just to walk away with this image, where if we do our job right, perhaps the human pangenome can just be a black box to clinicians. You don't necessarily need to see what's happening inside. However, the data input, maybe it's read data, short or long, or even assemblies. And as you can begin to select now your reference coordinates of choice. Maybe you want to work off of HG19, 38, the T to T genome, or coordinates that now bring in this extra substrate of new information we're getting from these more complete assemblies. And this last one I think is really important because we get a question about this a lot, and this is an active phase of work within the HPRC, where we do understand that there's a lot of common polymorphic sequences that deserve to be placed in a reference coordinate-based system. 700 megabases of non-reference sequences are greater than 5% of the haplotype. And we are exploring different methods right now, like RGFA was proposed by Hung Lee, which defines these additional sequences in stable coordinates. So we are thinking along these lines, and so hopefully that's something we can work together as a community as well. So in the initial pangenome, what we're talking about once again is 47 genomes. However, we were instantly identifying 120 megabases of new sequences in the human reference, 282 billion bases of haplotypes that were resolved. We have over 1,500 gene duplications and more than 20 million small variants. This is a small number of genomes, but a tremendous amount of information when it comes to understanding new variation. And this gives us access for the first time to some of these more complex regions. Here I'm showing you the HLA locus, where we can begin to study all of the different haplotypes that are in this region for these 47 genomes. If we were to think about another more complex medically relevant site, like the cytochrome P450 family of enzymes, once again, this is something I introduced earlier, but this one's particularly important because it's responsible for metabolism, about 25% of these clinically used drugs. And variation in this gene can affect how individuals metabolize these drugs, and multiple copies of the genes can lead to ultra-rapid drug metabolism, and others, if you have a slow or poor drug metabolism, then you have a different version or copies. So by having this type of structure, we can begin to look through each of the individual paths that go through the pangenome, and we can begin to make predictions on the count, the frequency, and even the haplotype that we observe on these different walks of the pangenome. We are expanding tools rapidly, although admittedly, it's early days. I feel like we're very excited to move into more and more of these long read mappers, but right now we're celebrating tools like Giraffe, Deep Variant, and Pangene. All of these have been previously published or open source and are available to everyone in this room. We also want to work towards improved representation of tandem repeats, and these are just tandem repeats in the genome, but also tandem repeats that would be closest to my heart, which are in the centromeric regions. We want to improve RNA-seq mapping, functional data sets such as ChIP-seq, and all of these types of data that we've celebrated with the use of HG38. We need to have a clear path of how to bring that information into the pangenome and have information that comes out. So while we understand that adoption of the pangenome will take time, and the pangenome methods, all the downstream tools are evolving fast, there are really promising applications of it right now. And so I wanted to at least put this plug in. We just went through our renewal, and there are now some new funding opportunities for anyone who's interested in actually getting funding to support pangenomic tool development. The application due date for this is November 1st, so it's coming up. But please think about applying if you'd like to be part of this team. So this is where I take another pivot. What I've been talking about so far is this dedication to complete reference genomes and the benefit of having complete reference genomes, and then moving into a largely NIH-funded effort to try to create a human pangenome with a limited focus of 350. But I think everyone on our team understands that this is a critical global resource and that it actually needs to be developed by and provide benefit to the broader global community. In other words, this should not be one nation, one group of scientists. This needs to be a federated global effort. And that's where we had launched the Human Pangenome Project, and that's where there's a celebration of it now being part of the driver project for GA4GH. Once again, with this idea of being a global resource, this is the same idea as what we had for the first human reference genome. It needs to be open access so that anyone around the world can once again download into their computer and then make use of these to make instant's not a word we use in genetics and genomics, but more, it would allow people to have access to basic research, expanding understanding of clinical genomics and genetics and understanding human histories. So this really is something that we need to come around as a global organization. We understand that this federated alliance of global partners need to have shared technical standards to ensure the resource is of sufficient quality. We need to absolutely emphasize these ethical and policy standards. We need to have responsible and secure resources for sharing, metadata, all of these that speak to these various working groups and work streams. We need to have federated systems for data sharing, and we need throughout it all, and I know this could be positioned under the ethical and policy standards, but just as a further emphasis, include care and fair principles in what we're doing. We are celebrating being a driver project. I think this is really where we are hoping, after my talk, to really engage with the community to ensure that we're able to be positioned to broaden our development and release and hopefully ensure global acceptance. We need to think about how we're going to start to increase our ethical and policy standards to ensure that we're respectful in terms of our domestic and international community engagement. And we need to start thinking deeply about how we're going to start using GA4GH technical standards in the work that we're doing. And along bottom, these are the champions for the Pangenome Project that I hope you see more in various meetings. This is emphasizing on this slide that the Human Pangenome Reference Consortium with the US flag above it is really defined by two major funding sources. This is the Genome Center, which is based out of UC Santa Cruz, and the Coordination Center, which is out of WashU. We hope to be just one of the teams seated on this global table. And this is what I'm trying to show you here with the outreach to each one of the foundational work streams that are here at GA4GH. It's hard for me to imagine which work stream we would not benefit from having some engagement with. We've already started to develop, and this is really crediting the wonderful work from Heather Lawson and our team and Angela Page as well. When we look at the Human Pangenome Project, when we've developed this international outreach working group where we've started to cultivate a community with GA4GH already. When we think about standards and development with GA4GH, we need to start thinking about guiding principles for these types of international partnerships, including consent, data usage, and data sharing. Our team right now has our head down trying to develop policy documents and start to engage with international collaborators and partners around the world. We're fostering an environment of scientific excellence. We really want to make sure that we're not talking about the number of genomes, but the quality of the genomes and the quality of the relationships and our dedication to ethics and moving forward with this project. We want to create pangenomic software ecosystems, and that will really broaden our user base. We think this will lead to improved genomic research standards, data sharing, and reproducibility. All of this, of course, has a home within a lot of cloud-based workflows that I think our team is working toward. And ultimately, what we're aiming for is to be a bedrock for clinical sequences and analysis. As we move into precision health, this is the reference genome that will take us closer to equitable healthcare. There's an acknowledgement here of, I think on the first day it was really fun to hear about the first white paper of where GA4GH is going. And I think with this slide, I just wanted to give myself maybe an opportunity to be a futurist too. And the idea that I think in the next 10 years, these T2T genomes are going to be everywhere. It's getting cheaper and cheaper and cheaper, and we get so much value from assemblies. What do you do in a world where you have assemblies now of all of these humans? And what happens now if we are sharing these pangenomic tools and individuals who have their own assemblies, their own cohort, are using these pangenomic tools to create their own pangenomes, to go through their own studies? How do we begin to expand the use of pangenomic tools and databases, not thinking about everyone coming into the pangenome, but thinking about pangenomes and how they engage with one another around the world? Ones that have different access of open access, closed access, data sovereignty models, on the cloud, off the cloud. All of these are big questions that I think over the next 5 to 10 years our team needs to think very seriously about. I also think that this is an opportunity to think what happens when you augment perhaps a private pangenome with the public pangenome? Are there opportunities for quality assessment in that space, and how can you trust the results of that work? So there are also perhaps important global partnership questions that we need to face as a larger pangenome project. One of the questions that I catch myself at least thinking about, and the kind of front of mind, is who benefits from this work? Which human haplotypes are being represented in this resource? How can we always keep that as our North Star for which we develop our tools as we do sampling, is keeping who benefits at the front of mind. Sovereignty, can we begin to think creatively about how to ensure that there are tools and ecosystems that benefit from different models of data sharing? I have emphasized open access for our reference resource. However, there is an understanding that we need to work to ensure that there are data rights and interests, for example, for clinical data sets as well as indigenous peoples, that need to be secured, and we need to start thinking about tooling in that space. We need to think about meaningful partnerships when we start to extend outside of our domestic partnerships that we have now. How can we start to organize with trusted partners that have long histories of trust within the communities that they represent? And how can we work across new ethical frameworks that perhaps are not as westernized or things that we're so common and comfortable with right now? There's a justice component, which has been an incredibly fun conversation I've been having with colleagues at UC Santa Cruz, thinking about who will gain from this resource. And this is bigger than just the pangenome team. However, these are questions I think whenever you take a step forward in a project this big and this impactful, that you have to take ownership in thinking about what you're doing in these contexts. Who will have first access? Is it going to be the clinical genomics here in the United States or in Europe or in the global north? How can we design new tools to run genetic analyses, and who's leading these new technologies? And at what cost? We always need to ask at what cost. How can we ensure that this historical mistrust doesn't get repeated over and over again through sloppiness or unintentional harm through our group? So I'll end my talk here, but really I wanted to emphasize the two main points that, of course, the Human Pangenome Project aims to broaden studies of genetic variants that can impact the effectiveness of medical treatments. We want to influence the risk of developing certain disease and also think about how we can be that bedrock for personalized medicine in the future. However, the legacy of this project may be a mixture. It might be a mixture of the tools that we provide, but also through the technical achievements and the accessibility of regulatory oversight and how we do outreach. And with that, I'll end my talk. Along the top, I hope you can see that I am representing an amazing number of scientists that represent a number of different institutes. On the bottom is our face-to-face scientific advisory board meeting. And once again, I have our company partners on the bottom. And thank you very much for your attention, and I welcome any questions.
Thanks, Karen. That was terrific, and you did spur a bunch of good questions. So okay. I'll just read them so you can, and then we'll try to do them in the order of voting, which will jump around. But if people want to upvote or add questions, please do so. First question: are you concerned about using cell lines? Are you introducing non-biological artificial variants that are telling you more about the cell line cultivation than the original human?
We absolutely were concerned about that at first. I can at least share with you that we do some pretty careful QCs in terms of every single cell line we work with. We do cytogenetics. We exclude about 12% because of chromosome abnormalities. We also do SNP chips. We also went through and we collected samples of individuals where we have their blood-based HiFi data as well as their derived LCL data, and we do a comparison, and we got a very boring Venn diagram when we were making our variant calls, which was very nice. And we're also working through Biomed to try to create this type of matched LCL and PBMC banking, which would allow for that comparison once again.
I'm going to take the next two together. They're related. They're a little bit different, but they're, the first is how do you decide who to go after next? You talked about it a little bit, but I think the question is probably asking for the next level of detail. Yeah. And then how do you know when you have enough, right? You're getting the next one, the next one, the next one. Is there an asymptotic curve or not? Do you see it? Can you predict it? What do you think are going to be the things that you'll find that are increasing or decreasing effects of scale?
Right. With a project of this impact and with a charge of being open access, when you start to do this type of prospective recruitment, it would probably be in anyone's best interest to do this carefully and ensure that you set this up in a way where you can really study this in terms of how you're communicating consent and how you're bringing people on. So although one could imagine that the diversity question may bring you to the farthest corners of the earth and trying to build partnerships there, our team for the next steps are really trying to launch prospective recruitment through our trusted partners at BioMe, where we've been working for 3 years to try to establish careful protocols, consents, and thinking through screener questions and things of that. I think that these types of international partnerships, as we start to leave the comforts of domestic biobanks, will once again happen through these very meaningful, trusted partnerships to where we're not just going chasing helicopter science and diversity net per se, but really trying to work through community-based efforts to ensure there's some element of respect and benefit sharing. So in some ways, that's a little counterintuitive to the scientific request of just chasing the variants, but I think I want to emphasize that type of careful outreach in the world that we're living in and the resources that we're building that are open access. With that being said, I think that this over the next 5 years will absolutely be iterative. There is a call here for benefits, as I mentioned on my slide, being point number 1. If you are entering into a clinic, I don't care where that pin drops on the globe, if you can't benefit from the Pangenome reference resource, we're not doing our job. So it has to be at that measure of when someone walks into a clinic, do they have a haplotype that represents them by so many meioses away to where they have informed information by the use of the Pangenome. Now, where we get there, of course, will be through constant sampling. Like I showed before, we're in early days of doing this, but trying to keep hold on, where we, through various data sets, we've already teamed up with Nomad, we're expanding out into the All of Us consortium, we're trying to think about how we can broaden this type of diversity estimates for sampling.
If I could follow up on that and ask a much simpler and more quantitative thing. T to T was 1,
yes.
The next paper was 47. What are the next 2 numbers in that sequence? 1, 47, and yeah.
I almost put a slide up, but I don't want to get people caught up in our versioning. So we have our draft release. The next one that we're doing have roughly 150 genomes. I want to emphasize that we prefix with near T to T. Reaching the type of T to T genome that we have for CHM13 is not an automated process. It takes teams of experts to pull it to that level. So we're just not there yet, but these near T to T genomes are amazing. So that would be 150 or 300 phased haplotypes. And where we're trying to go is the 350 or 700 phased haplotypes by the end of year 5, which would actually be the end of next year. So we're trying to create a new version to release once we reach kind of the throughput of our first phase of our initiative. And as you can imagine, over the next 5 years, if we're fortunate enough to get our renewal, then we will be able to extend that out to where we're aiming for 550 or over 1,000 human haplotypes.
Great, and that's a perfect segue to the next question, which the kind of mess one, which I'll let you decide if you want to treat this as a yes/no question or say more. Given the kind of mess we have around stable identifiers for the current reference genomes,
Yeah.
Do you have any thoughts about how you're going to do something better than HG38++ for identifying future references?
I feel like this is a moment where I should call my colleague Benedict on the stage. But I definitely feel like everyone's terrified of using the pangenome that I talked to. I think there's a real benefit for our team to demonstrate that, hey, if you have an assay that's kind of concentrated on HG19, the pangenome, you can get those same coordinates and you get this advantage of more. And I think that that's the same for HG38 and the same for CHM13. Once we start moving into these new coordinate spaces, the ones that I professionally care so deeply about, I also am the first to admit that we don't have a lot of functional data over these sites. We don't have extensive Nomad allele frequency information. We don't have a lot of information about how they correspond to disease. So I think that these early days of trying to bring in these new sequences will really kind of be an engagement for innovative thought leaders that want to really explore in these unexplored corners of the genome. And through that process, hopefully we'll come up with a really standard format to bring those functional elements they're finding into a place where people can really use them in a coordinate-based system. It's not a great answer, but I think that this is a place that we take very seriously right now, and we just don't have a clean answer, but maybe if you invite me to your next plenary, I'll have an update at that point.
Right. We have covered this next one, but I think, I'm going to ask it and then guess my answer, your answer, and then have you correct it. What's enough, right? If it's 147, 150, 350, when does that taper off? Will there ever be enough individuals to cover full diversity? Are you able to predict, yet, the unseen features based on, and my guess as an answer is, doesn't matter yet. We know we have to get to at least 350, and then we'll figure it out. But I'd be fascinated if you think you see what that curve looks like beyond that.
Right. I think that there's a goal that was stated in our first phase where we wanted to cover global common haplotypes at 1% allele frequency. And with that metric, it makes it easy as a task for us to start to evaluate broader and broader and broader databases to see how we're performing with this goal. However, what we're leaving off is really some of these more complex structural variants. And I think as we start to make more partnerships and we start to build out into understanding the allele frequencies in these more complex and dynamic regions, that may change how we build that slope. And so there are these kind of technical components that our team is working on now. Right now we're really focused on SNVs because that's what we have clearly accessible to us with these large biobanks. With exome sequencing, we're not going to have long-read data from tens of thousands of individuals. However, we are thinking along those lines as well. And where we ultimately want to go, as I mentioned before, it might be at the end of 10 years, but this is going to be an iterative process where we start to figure out where are the holes, who are we not serving in the world, and how can we update the pangenome in order to reflect these holes of service or who's not benefiting. And I think that will likely even outweigh these early kind of counting of haplotypes.
At the risk of breaking a metaphor, the classic of looking for keys under the lamppost, and your first thing is, well, let me light up more of the street.
Yes.
At the same time, you're also adding ultraviolet light, and now there are new holes in what we can see that you didn't even know existed, and so.
Yes.
Yeah. That's great.
We've used unknown unknowns in corners, which no one wants to repeat, but here I am on stage. Yes, but I definitely think that our team acknowledges that there's, as we branch out, this has to be iterative because the databases, we've already celebrated at this conference, are growing at such an exponential rate that this information hopefully will gain as we move forward as a consortium.
Right, the next question is, I think, an excitingly ambitious question saying, yeah, okay, you've solved DNA. What's the state of mapping transcript isoforms from long-read RNA sequencing to the pangenome? And where are the streetlights missing in transcriptomics space?
I would encourage to read the draft pangenome. I think there's a really lovely section where we're starting to work with RNA-seq mapping. Also, from Nick Payton's group, there's been developments there of looking at RNA-seq in separate papers. So we are developing tools in this space as well. I think where we're wanting to move is into these long-read isoform. I think that was a very important part of this question of how do we start to bring in these really important long-read data sets, and that's at least coming from our face-to-face meeting, I think there's a lot of reasons to be optimistic that that's on the horizon.
This next one you may have covered. If you have more to say, how do you prioritize which genomes to tackle first?
I think I've already emphasized that, but I'll say it again because I think it's so important. I think that we're operating through prospective recruitment with these very careful measures for community engagement, IRB approval processes, and ensuring that we're doing this in a way that is really onboarding a lot of ethical considerations.
Which is a great segue to the next question. Can you say more about how you'll incorporate GA4GH, both technical and ethical policy standards? So this is really the next level of detail. What do you think, you're a driver project, where do you think you're going to drive?
Well, as I showed, when you ask me, I'm like, I don't know where we will not engage in terms of the existing work streams, but ones that I can clearly step up and say we're already starting to think about and work closely now is with international partners. We are collecting consent documents. How do we begin to go through a process of harmonizing consents? How do we begin to think through these very distinct policy questions and guidelines for what an international partnership in terms of ethics and standards needs to look like? This is something that I think is very front of mind for our consortium right now as we take our first step forward. I think as we start to think deeply about cloud-based resources and workflows and how we're going to engage with existing partners from the GA4GH community for trying to pull the pangenome into what you're doing and also think about tooling that needs to be established to make that happen. I think that that's something else with our pangenomic tool community. How can we start to think about reference mapping and variant calling and move into that? So we kind of see those two major tool developments, I know is so broad, but really when it comes to anything that's reference-based variant calling and cloud-based workflows, and then with the ethics, I think that will be an incredibly huge value to our team as we start to move out of the comforts of domestic domestic partnerships and more firmly into trying to create something that's more global.
The next question is both drilling into a specific potential example, but I think I'll generalize it. Is GFA a future GA4GH standard? Is some community discussion needed? And I'll generalize it to, if you picture plenary 2024 or plenary 2025, what do you think are 2 or 3 new GA4GH standards that you would like to see have resulted from your new driver project? Either this new capability of standard X, or perhaps, as with the question, here is a new GA4GH standard.
I don't think I'm the most appropriate person to answer this, and so I want to emphasize that folks should speak with my colleague Benedict after my talk. However, I think that—
Benedict, could you raise your hand just so—
there he is, good. Yeah. Let me embarrass him over here. But I do think that, at least from the conversations we've had within our Pangenome working group, deep thought has been going into, GFA and graph formats, and I think that that's something that we would love to engage with this particular team, thinking deeply about how, if GFA is that model, or if we should consider other formats. This is something that I know I've heard mentioned several times, even by Heng Lee's group. I think, once again, this idea of how you take 2 GFAs and you start to look at quality assessment measures, that's something that is actually quite important for a couple of projects that I'm deeply involved in right now. And so I think that thinking through those types of standards and work streams for comparing the quality assessment of pangenomic tools and reference databases is something else that I'd love to see some progress on.
So the action I'm hearing from that is, for anyone here who's responsible for a standard or a working group, and you're not sure how this will affect you, reach out, is what I'm hearing. Because I think the answer to, is community discussion needed, is absolutely yes. That's the point. That's why.
Yeah, we've had our heads down, and I really want to credit, it's a draft, and I think that that was a very humble title, but it was an amazing accomplishment to put out the first pangenome. And I feel like that was a small step forward, but we're now walking faster and faster, and I think that we've had our heads down now trying to figure out these technical components. How do we do T2T production, which is not an easy task? How do we do QC? How do we build a pangenome for the first time? And now I think we're at this really exciting pivot where we start to think, how do we start to create tools that people will use? How do we start creating cloud-based, workflows? How do we begin to engage with individuals in the clinical genomics space? All of this is something that I think is really building from the success of our first phase, where we really had our heads down looking at technical components.
All right, we have about 2 minutes left, so let's do lightning round on these next few, each of which could be a 10-minute conversation. Okay. Computational complexity. How does it scale with the number of genomes? Is twice as many genomes twice as hard, 4 times as hard, 1.5 times as hard?
I took out the slide that I should have kept in, apparently, but there is, of course, as you start to add more and more genomes, there is an opportunity through shared haplotypes for that to actually have some advantage with compression. And our team has already started to look at that with the few pangenomic databases that we've constructed in these early days, but we're hoping to go through and actually see where those pressure points are and test as we start to move, as you mentioned, into this 300, phased, 700 phased haplotypes start to scale up. I think that our team will have even more to report in the near future.
Right. Is there a concern from American Indian tribes about, and I'd say this is probably one of about 100 potential cross-references between pangenome and social and ethical and societal and identity questions, but have you heard or are you in discussions around this one in particular?
This is something that I think our team takes seriously, but we never feel like we're at least I can speak for myself. I feel like there's always so much more to do. I feel like what we're in the process now is, how do we organize and start learning from the past, start working with teams like the All of Us, think about how we can improve consulting, think about how we can start to form early partnerships. This doesn't have to be necessarily just with North America or South America, but even internationally with groups that have different views of data sovereignty and data ownership models, and how can we begin to operate through different ethical frameworks? How can we begin to think about how the communication of the project could be improved? How can we start thinking about, benefit models and work with trusted partners who have long-standing history of working with groups and representatives and thought leaders in the American Indian tribes, or Native American tribes as well?
All right, as the last question, and the answer might be call me, but how can international efforts take part?
This is a very timely conversation. What we're trying to do at the beginning, it was how do we do this in the most broadest way possible? How, you don't want to just call all your friends from your network into a closed room. And so the idea that we proposed was to develop a policy paper, one where we say, hey, this is a Human Pangenome Project. As I mentioned on one of my slides, no one group can do this. This is a global resource. It needs to be developed globally. It needs to benefit globally, and kind of put that on page. Create this as a bioarchive or something that's in development, and then reach out and start doing workshops and take information, bring in partners, bring in collaborators, redevelop that statement, that white paper, with new international partnerships, and then work with GA4GH to start thinking about how to organize that type of federation, as I showed in my slide, where we're just one of the members sitting at the table.
Great. Thank you very much, Karen.
Thank you.
The questions that haven't been answered will be on Slack if people want to follow up. All right. So we're going to flow right into another 3 fascinating talks. We're going to do questions pooled for these. So after Angie and the next couple of speakers talk, we'll do all the questions together. So our first speaker is Anjene Musick. Angie is the Director of Scientific Data Strategy in the Division of Medical and Scientific Research and the Genomics Lead for the NIH's All of Us Research Program. Before joining All of Us, Angie was Chief of the Genomics Research Branch in the Division of Neuroscience and Basic Behavioral Science at NIMH, National Institute of Mental Health. Angie earned her MPH and PhD in genetic epidemiology from Johns Hopkins Bloomberg School of Public Health. And I have the privilege of working with Angie on All of Us and have been in a bunch of meetings and have heard some of, and I'm looking forward to hearing more about the latest of All of Us' work with long reads in particular. So take it away, Angie.
Thanks, David. It's really a pleasure to be here. It's my first GA4GH meeting, and it's been a lot of stimulating conversations going on. So my head is buzzing with lots of ideas. And I have a lot to cover in this talk, and I could not have asked for a better introductory talk than Karen's talk explaining about the value and value add for long read sequencing. And so I'm excited to have the opportunity to talk to you today about the work that we've been doing with All of Us. And let's see, my green button. Okay. So first I'll just start with very little bit about the All of Us research program. Hopefully most of you are familiar. But basically, we have this mission to accelerate health and research and medical breakthroughs by enabling individualized prevention, treatment, and care for all of us. My core mission, personally, my role in the program is on that far right, which is deliver one of the largest, richest, most diverse biomedical data sets of its kind that will be shared broadly and made available to all and be secure. That's really what I do day in and day out, is think about the researchers and what is going to really push the needle forward in novel discoveries and reducing health disparities. So, in a nutshell, the goal of the program is to enroll 1 million or more people from across the United States. It is not intended to be representative of the United States, but just to cover the diversity of what we experience in the United States. Currently, we have data available for over 400,000 participants. The number, what I would say, one of our most important core values of the program is the focus on diversity. About 3/4 of our participants are in what we categorize as underrepresented in biomedical research, and 50% of those are from racial and ethnic minority groups, i.e. non-European descent. And this is, I think, one of the biggest differentiators of our program and what makes the genomic data in particular very, very exciting and really going to create a lot of new discoveries. Okay, so our core protocol is, so because we treat participants as partners and all of our activities or data donations are optional. Participants can sort of choose their own path. We have a modularized consent process. So there's a main consent, then there's consent to share your EHR records. There's a consent to get DNA results if that's what you choose. So our core data set is really some surveys that people do. We do some primary physical measurements. We ask participants to provide blood samples. If they are unwilling to do blood samples, we'll collect saliva. And then they also have options to donate things like their Fitbit data. And we've also added a new data type as of 2 weeks ago, where we're doing some cognitive tests, some interactive cognitive testing. Okay. I'm just going to talk briefly about the timeline of this program, because I think this is one of the things that I'm most proud about and get excited about, is how much we've delivered in such a short amount of time. The program was instantiated in the 21st Century Cures Act in 2015. Our first funding announcements went out in 2016. We did not actually, so our national launch was May of 2018, so we just celebrated our 5-year anniversary. And we did not actually start analyzing DNA until 3 years ago, in 2020, for a variety of reasons that are not important. But here we are. And 18 months later, we delivered our first genomic data set, which was 100,000 genomes. And then in this past April, we released our second set of data, which was close to 250,000 genomes. All right. I'm going to rush through some of these slides, but we have 3 tiers of data, and I would encourage any of you who are actually on your computers to sort of check this out as I'm talking, and you can keep me honest with some of these numbers. But our public tier of data is something that I personally use all the time to double check on numbers, but it will give you summary statistics of all the data that's available in the researcher workbench, so that you have an idea of if there's anything of interest to you that may support your research questions of interest. So you can see what different EHR domains we have, what the genomic data is available, the different surveys. You can download PDFs of all the survey questions. You can see the summary statistics of all the answers. You can see here in the little box under genomics this is our short read SNP and indel data. We have 245,000 participants with over 1 billion variants that are searchable in our genomic data browser. Again, just to emphasize, this is all data that is currently available in our researcher workbench for registered users. What we added this spring was, in terms of the genomic data, was some structural variant data on the short read sequences, and what I'm going to talk about more is our long read sequences. One of the things we try to do as a program is not just dump a bunch of variants into a bucket and say, have at it. We want to try to make the data as usable for researchers as possible. So things that all researchers will do. Doesn't make any sense for everybody to repeat the same introductory cleaning QC steps. So we do things like running the PCAs for ancestry, relatedness. We create all sorts of auxiliary data just so that researchers have even more resources and more curated and refined data sets to streamline their analyses. So we're doing the structural variant calling, which is very computationally intensive for all of the short read sequences. And so this was just our first tranche of that data. We'll be having our next release of that data sometime probably in January for 100,000 participants. Okay, and then here's just a quick glimpse of our genomic data browser. So you can go into, you click on genomics, and then you can click on, choose your variant or gene or region of choice, and you can see how many variants we have in our database. And it's broken down just with the similar categories as nomad in genetic ancestry distributions. Okay, let's get to the long read sequencing. So I don't think I need to go into why we want to do long-read sequencing. One of the goals of the program, I mean, obviously it was conceptualized in 2015. We knew that technology was going to change. We knew we would want to keep up with whatever new things would be coming online over time, and just keeping an eye on things and having the ability and the flexibility to pilot certain new technologies as they sort of bubble up and think, okay, let's just take a look at this and see how it's working. So the long reads is one of those opportunities that came in, and as Karen very clearly demonstrated, there's just huge chunks of the genome that are missing with short read data, and especially in medically relevant genes. So there's no dispute, or nobody's saying, no, that's a bad idea, we shouldn't do that. So away we go. Again, it's just, we know that these large structural variants probably have much greater effect on disease and health conditions. Even though they're more rare, they are more likely to have effects on gene expression. Okay, this is a busy slide, I know. And I'm gonna talk a little bit, try and break this down a little bit, because it is complicated. So the main thing on this slide, so we've sort of been working in a phased approach. So our first award went out in late 2019 to Hudson Alpha to do some technical development on these emergent technologies for long-read sequencing, and just do some technical work, not using participant samples, but using other reference samples and comparing different technologies so that we can get a better sense of where these technologies were going, what would be feasible or even plausible for scaling at any scale. So we do have a paper that is under review right now, just analyzing those technical replicates across 3 different technologies, and really focusing in on capturing the medically actionable genes that are not well captured with short-read sequencing. And I'll talk more about these other pieces as I go. So phase 1. So what did we release in April? We released our first 1,027 whole genomes with PacBio at 8x coverage. So given that this was a technical development project, we didn't know going in what we wanted. So there was a lot of things going on. The protocols were changing. There was a lot of back and forth. And so we finally just reached a point where it was like, okay, we'll just take what we've got right now and let's just go with it. And so that's where we ended up. So we have been working tirelessly on QCing this data and developing and releasing the relevant data for that. And we're working on a marker paper to describe that data as we speak. This was announced in April when we released our last data set, along with our 250,000 short-read genomes. And the long reads are a subset of the people who have the short-read sequencing as well. So for this first phase, we started back in 2019. We hadn't even touched a DNA sample in our biobank yet. We said, what's an obvious place to start? And everyone, we have a large working group. When I get to my acknowledgement slide, you will see how many people have been working on this effort across the consortium. And the consensus was, let's look at African Americans because we want to maximize diversity and new discovery of genomic regions. So that's what we did. So the PCA plot you see is actually what the full data set looks like. And then in the light green, sort of in the bottom corner, is the African American samples that were selected for this pilot. Okay, we're gonna move on. So I think one of the main takeaways I want to make sure it gets through is, given that this is all very new, we are literally developing methods as we are analyzing the data. It is very, from week to week, we're having discussions and talks and exploring and tweaking things and figuring out what's best. And so there is no consensus on what is best right now. So what we have done is we've aligned to 2 reference genomes. We've done the HG38 no-alt as well as T to T. And on we've also done de novo assemblies with the GFA format, so that's a common theme. And then we've done multiple different workflows for structural variant calling. We've done phased and unphased single sample SNP and indel calls as well. So there's a lot of different variations of how we've curated this data and made it available. Okay, I'm going to be running out of time, so I'm going to go quick. So just we've already identified out of this 1,000 millions of new structural variants. A lot of them look like they're high impact in terms of function. One example that I wanted to highlight is the LPA gene, which has been historically known to be difficult to capture with short read sequencing. We've identified 25 novel exon overlapping structural variants in that gene in this one data set. So there's clearly a lot to be mined here. All right, so phase 2. So again, when we first started this technical development project, we didn't know where it was going to take us. And once we started to see that we were actually getting good data and the technologies were getting better, the prices were getting better, things were becoming, we were like, okay, let's do more. So that's what we're doing. So the goal is to enhance discovery of pathogenic variation, to really construct a large structural variant resource for genotyping and imputation into other data sets, sort of build out a nomad of such for long reads, that kind of thing. And yeah, just develop a more comprehensive database of genetic variation from these long read sequences. So we took a all-comers approach with not choosing any one technology and said, let's just do some head-to-head comparisons as well as not necessarily a bake-off, but just see where these different technologies will go. And so we're doing a whole hybrid mishmash of different strategies. Okay. So this is, again, where we get to the complicated slide. So on the left was the technical pilot. So that manuscript is under review. The phase 1 pilot, which was released, it's out in the public, it's out there. We're working on that manuscript right now. Phase 2 is in full force, where we've now expanded to all of our other genome centers, including Broad Institute, University of Washington, Baylor College of Medicine, and Johns Hopkins University, and doing a variety of different approaches across both PacBio and ONT at different read depths. So that's kind of it in a nutshell. Our next data release will be in the fall of 2024, and we anticipate that we'll have approximately 2,500 PacBios and about 650 ONT genomes, and all of our data production is scheduled to be completed within the next 10 months, so then all the remaining data will be made available sometime in 2025. And this is my acknowledgments. There's a lot of people doing a lot of work on this. This has been, and everybody seems to be having fun. This is really, it's a fun project. Everybody's like, oh, this is really cool. I never knew, you know? So, it's made me feel like a scientist again. It's really fun. And I really mostly want to thank Kieran Garamella, who has been leading this effort. And it's just been phenomenal to work with. And I used most of his slides, so I just have to thank him. Anyway, I'm over time. Thank you very much.
Thank you, Angie. We'll do the questions at the end. Appreciate that very much. Anshul, come on up. As Anshul's walking up, I was thinking that we've heard a bunch of presentations on more data, and I think more data comes with more signal and more noise, and that means that people who are trying to find signal and noise become more important. So Anshul Kundaje is an associate professor of genetics and computer science at Stanford, The Kundaje Lab contributes to the collaborative development of foundational genomic and molecular data resources in several consortia, including ENCODE, Roadmap Epigenomics, and IGVF, specializing in developing interpretable deep learning frameworks for decoding regulatory DNA and genetic variation. So, Anshul, I'm looking forward to hearing what you have to tell us.
Awesome. Can you hear me? Okay, awesome. So thank you so much for the invitation today. Seems like a really great meeting. I loved your keynote today. So I'm going to switch gears a little bit, as David said, and move a little bit into how we can leverage machine learning to try to understand DNA sequence and also genetic variation. And the fundamental question I'll try to get to today is now that we have thousands of genomes and pangenomes and all kinds of genetic variants, how can we accurately, effectively, and efficiently understand the consequences of these variants? And I'm going to focus on a specific class of variants today. As you know, there are variants that affect protein coding sequence and also non-coding variants. There's actually been excellent work in the coding space. In fact, yesterday, DeepMind released this really nice method called alpha-missense, which seems to be pretty state-of-the-art. But as you know, a large proportion or a large majority of genetic variants associated with disease are in fact non-coding. They don't directly disrupt protein coding genes, and they happen to be enriched in regulatory DNA. So my talk is really going to focus on this component. I think also on the clinical side of things, there's obviously lots of good standards for moving coding variants into clinically actionable situations. The non-coding space is a completely free-for-all. Most people just discard those kinds of variants, and so there's a lot of work to do in this space, and I think machine learning potentially has a big role to play here. As you know, if you're training machine learning models, you need data. This is the kind of data we typically work with. Over many years and decades, many large projects have generated massive amounts of molecular data, and you can think of this data sitting in a sort of a three-dimensional cube. You got one axis, which is genomic coordinates. You got another axis, which is different kinds of molecular readouts. For example, transcription factor binding profiling, chromatin accessibility, histone modifications, all kinds of molecular markers of regulatory and also transcriptional activity. So RNA-seq, nascent RNA, and so forth. And then each of these assays is typically performed in some amalgam of cell types, tissues, and now with single-cell technology, you can also do this at the resolution of single cells and in multiple individuals. So this teracube of information is incomplete, but it is absolutely massive. And it's a great opportunity to take this kind of data and try to understand the fundamental question about how 3 billion base pairs, or give or take, how they actually encode this function and how they actually give rise to all this beautiful context-specific dynamics. And so I'll focus on those two components. And the workhorse models that we typically use, I'm going to focus mostly on my work, but I'll try to give some context of what's happening in the community as well. We've released some models a few years ago. These are the first of its kind. They are base pair resolution models. So what I mean by this is, think of these models as like text-to-speech converters. The text is the DNA sequence. The speech signal is the molecular readout from some assay. Could be transcription factor binding profiling. It's a sequencing experiment. And you get read coverage profiles at single base pair resolution. Typically, what's done is this data is heavily processed. You do some kind of denoising, some statistical analysis, calling regions of enrichment. So you're doing a lot of heavy processing beforehand, and then feeding this to a machine learning model. Now, what deep learning models actually excel at is really mapping complex inputs to complex outputs. And so what we thought is you might actually not need to process the data at all, and instead leverage the power of the model to do all the processing for you, that is learning the biological signal and extracting biases, all the noise and the spurious signals that you don't want to learn, right? So our models actually take DNA sequence and map them to single base pair resolution raw read coverage. There is no processing at all. There's nice advantages to that. You can literally just map your reads, get a BAM file, get a coverage track, and that's it. You're ready to go. There's no need to worry about which peak collar should I use and which this thing and that thing should I use. There are, of course, dangers of doing this, but I will show you how, if you do it carefully, it can be extremely powerful. And so these models just basically learn mappings from DNA sequence to molecular profiles, single base pair resolution. But when you think of deep neural networks, you often think of these uninterpretable black boxes, right? But we've spent a decade actually building a suite of interpretation tools, and I can't get to all of them. Hopefully, Suin is also a world expert in this space, and she will talk a lot more about explainable AI in her next talk. But I'll just give you one flavor, which is once you have trained a model on the data, it's very good at mapping sequences to profiles. You can, in fact, interpret these models. You can use them as oracles, and you can have them tell you, if given a specific sequence, genomic or synthetic, which base pairs in the sequence are driving the activity. That's a very fundamental question. And this can be done very efficiently. We have lots of good methods for it. Some of them are listed here. Others have other methods. But broadly, I'll just give you one really cool example of how the power and the danger of using neural networks, but also how you can take some of the Achilles heels and turn them on themselves and make them a power. So you'll understand what I mean when I go through this slide. What I'm showing you here at the top is a specific segment of the genome, and these are base pair resolution coverage profiles, read counts, coming from an experiment called ATAC-seq, which allows you to measure chromatin accessibility in the genome. These assays will highlight regulatory DNA. And you see these beautiful, these coverage tracks, and you're seeing this kind of squiggly profile. If you train our neural network models on this data directly, and you predict what the sequence underlying this region how it would generate an ATAC-seq profile, and note that the model is not trained on this sequence, it's trained on a subset of chromosomes, and we're making predictions on completely unseen sequences. The second track that you see right there, that is a prediction from the sequence model. The model just has looked at sequence, and it's making a prediction. You can see they're almost indistinguishable. And this is not a cherry-picked example. In our paper, we actually show that the performance of the model is at replicate concordance. So if you did 2 replicates of the experiment, how similar they are, this model can predict the profile at single base pair resolution with exactly that kind of replicate concordance. Now, when this looks great, right? We're done. The model's awesome. Let's interpret the model and see what it's doing. You see the little base pairs logo thing, that's the importance track, and the height of the letters corresponds to how much they contribute to the signal. Now, if you've looked at these kinds of signals sufficiently, which I have over 10 years, you see something in the middle that makes sense to you. That's the motif of a transcription factor called CTCF. It's a big protein. It binds and creates a massive footprint. Okay, and that's what you're seeing. That's what you should expect to see here. But the model's learning all this other crap around it. Like there's all these other bases popping up and down, and often people will say, oh, that's just noise from the neural network. Well, actually, it's more interesting than that. So the thing to remember is that every genomic assay you perform is not actually measuring just the biological ground truth. It is a surrogate measure. There is the ground truth biology that's affecting the readout, and there's the technology, the enzymes you use, the protocols you use. And so the observed signal is actually a mixture of the true underlying latent biology and all this other assay spurious stuff. The beautiful thing is we can actually leverage neural networks, training on the same data to dissect the underlying enzyme bias, and we can predict what the enzyme bias would look like for this sequence. And this is what it looks like, the green track. Now compare the green track to the purple track on top, and you will notice how similar they are. The bias of the enzyme that's used is remarkably similar to the actual observed signal, right? So this particular assay is very powerful, but it has a very strong bias. Now the beauty is we can regress out this bias and look at the underlying latent biological signal that was hiding in the data, and this is what it looks like. The blue signal is the underlying footprint of that transcription factor. When you subtract out the bias, that's what pops up. And this is what a neural network can do. It can take your raw data, which is a combination of the underlying signal you want and the other garbage, and you can deconvolve it beautifully. You can impute the data, the latent footprint at single base pair resolution, and once you look at this bias-corrected data and you interpret what the model is actually predicting in terms of the sequence nucleotides driving the signal, you just see the CTCF motif. That's the pure signal that you pick up. And not just that, but you see the little things on the right that I've highlighted? Those are repressors. Those are completely hidden if you had not corrected for the bias. Right? So you see the power and the danger of using a neural network. If you blindly used it and you looked at your first 2 profiles and you thought, this looks amazing, let's just go ahead with it, you would have been learning the underlying sequence drivers, the biological drivers, and a ton of assay bias simultaneously. But you can still use the models in exciting ways to regress out this bias, figure out the latent footprints, and also interpret the models and figure out precisely which base pairs are driving the signal. And just to sort of showcase again, you can, as I mentioned before, the alternate approach would have been to pre-process the data heavily and then train a model on that. So people have previously built statistical approaches to bias-correct attack-seek data. But when you train the models on those statistically corrected bias-corrected data sets, you actually don't correct the bias. If you train a neural network and interpret it, it's still learning the bias, right? So you can see how a neural network is an end-to-end framework, and so it can optimally perform all steps of the process. And if you avoid doing pre-processing beforehand and set up the network so that you can do it while you're learning, you can actually do it much more optimally than a stage-wise process, okay? So that's like sort of one genomic region in one cell type with one experiment, but we can do this in much more exciting systems too. So if you look at differentiation reprogramming systems, you have beautiful continuous dynamics of cell state transitions. Here's a two-dimensional map of single cells across a fibroblast to iPSC reprogramming time course. And we can train models now not just on, on a single cell or a single cell type. We can train it on all possible cells in this beautiful trajectory. And we can take any gene locus. For example, here's an ANOC gene locus. And what you're seeing, the tracks from top to bottom are the attack-seek tracks starting from fibroblasts to the end-state iPSCs across the reprogramming trajectory. And you can see how the signal that you're seeing is chromatin accessibility. When you see these spikes popping up, those are regulatory elements becoming active. You see this amazing, beautiful dynamics going from NANOG, which is a classic pluripotency gene, being completely off in fibroblasts, to progressively parts of the regulatory landscape turning on. And in the end, you can see the landscape is very active, right? Now, using these models, we can train them on all these profiles at single cell resolution now. And you can see the beautiful dynamic tracks that you can take any enhancer, any regulatory element in the genome, and trace across time how the sequence is being utilized by various transcription factors to drive that signal. So you see this kind of really beautiful combinatorics happening. There's an OX-SOX motif on the left that pops up midway through the time course. There's a KLF motif that pops up earlier, but then disappears in the last time point. And there's an OC motif on the right. This is all coming completely out of a black box neural network, right? It's learned all this underlying sequence syntax of regulatory biology. So this is really cool if you want to understand basic gene regulation. But another powerful aspect of the models is they are predictive. You can push any sequence into them, and they will predict what happens in terms of the assay they were trained on in the cellular context that they were trained on. So here I'm showing an example of how we can use the models to predict effects of variants without the model ever having seen genetic variation. It's trained on the reference genome on some single sample, and now we are in silico simulating what a variant might do, right? So think of taking a region in the genome, pushing the reference sequence through the model. Here's what it looks like. There are three models I'm showing you. One is a SPY1 protein DNA binding model. It was trained on SPY1 ChIP-seq. SPY1 is a very important transcription factor. The second is chromatin accessibility, and the third one on the right is histone modifications. These are predictions from the model at single base pair resolution. They're beautiful. If you look at the raw data, it's like super crappy, sparse, and noisy, and the model is able to impute the signal at base pair resolution. Now, all I'm doing is taking the same sequence and flipping the central base from a C to a G, okay? And you see what happens. There's a massive amplification of the signal. Let's just go back and forth. All three profiles, you can see a really beautiful amplification of the binding profile, amplification of the accessibility profile, and an amplification of the histone modification profile all the way up to 6 kilobases. So a single nucleotide change has a massive impact. Think of it like a blast radius of sorts, where the molecular profiles are reacting to that single base pair mutation, right? But we don't have to stop there. We can, again, use the model's ability to interpret sequence, and we can tell you what the variant is actually doing. In this particular case, it happens to be, when you switch the allele from C to G, it creates a beautiful, really strong motif of the SPY1 transcription factor, which interacts with the weaker motif on the left. They cooperatively, they cooperate together to amplify binding, amplify accessibility, amplify histone modifications, amplify gene expression. And these are validated, QTL across the entire chain of molecular events. Now, here's an important point I want to make. Our models are very cheap. They're not expensive. You can train these on 4 hours on a regular GPU on a single track, okay? And You may hear in the deep learning literature that scale is everything, right? Like, let's just scale up models. Let's build like giant transformer models with 70 billion parameters. Well, people are doing that, and companies like Calico and DeepMind have trained such models. I'm happy to say that our tiny little model that you can train in 4 hours beats those models very comprehensively in all benchmarks. Now, I'm not saying this is always going to be the case, but I suspect that in a large number of cases, if you're clever about how you build your models, and you think carefully about how you correct for assay biases, how you precondition them, how you set up your training data, I believe that small models will often be able to outcompete or at least play as well as the big models. I'm not going to go through these benchmarks, but these are precision recall curves, and the important thing is these are benchmarks that we derived from their publications. So we're not cherry picking our benchmarks. We're actually applying our models to benchmarks that they have in fact run in their papers. We can use this to fine map GWAS loci. So this is a very interesting application, not just QTLs, but sorry, the text up there is kind of messed up. But we're looking at a particular locus which has a significant association with coronary artery disease. And what's really exciting here is we had a beautiful data set of single-cell chromatin accessibility in a mouse model of atherosclerosis. This is very difficult to get in human samples because you can't really feed a human high-fat diet and trace them over time, but you can with a mouse. So you can get really beautiful rare cell types and cell states. And we can train models in mice. And even though now we're training on a mouse genome, on mouse accessibility data, once the model is trained, it's just a sequence to accessibility model. We can easily apply it to human sequences, right? So what we've done here is taken a GWAS locus, and we've used the model to score all the variants that are in that locus. And here's what the model predicts is the causal locus, causal variants. In fact, it's not one variant. It is 2 variants in perfect LD. You can see the beautiful haplotypic coordination. On the left is a variant which, when you have the A allele, you see that it produces this beautiful AP1 activator motif, and the C allele does nothing. When you swap to the other haplotype, the first variant has a G allele, and the G allele destroys the activator, and the second variant has a T allele, and the T allele actually creates a repressor motif. So the 2 haplotypes, you have a coordination between 2 variants right next to each other, where you simultaneously create an activator and destroy a repressor, or you create a repressor and you destroy an activator. And when they coordinate together this way, the enhancer goes from totally on to totally off. There's a massive impact, right? So there's all kinds of really exciting discoveries you can make. I'll skip this in the interest of time, but we can apply the models not just to common variation, but also rare and de novo mutations. So this is a paper we published last year in Cell. We looked at single-cell ataxic data from very early fetal heart development samples. Human fetal heart is the first of its kind. We built the same kind of models, and we use the models to score de novo mutations from case control studies, the PCGC cohort, children with congenital heart disease. And we were able to make really nice predictions and validate many of them, not just effects on chromatin expression, but also phenotypic effects on like tube formation in arterial endothelial cells and so forth. So with that, I'll just stop and say democratizing these kinds of models is very important. We've built one of the first model zoos for genomics. And I'll thank my lab and all our collaborators and our funding sources. I'll just leave you with a few thoughts. Trends, hype, and potential. Is scale all we need? Do we need to just build bigger models with more data? I have a lot to say on that. Is self-training all we need? There's exciting new approaches coming out, training these DNA language models and single-cell foundation models. And then I'll leave this slide just so you can take a look. I don't have time to go through it, but there are many interesting challenges and opportunities that maybe we can talk about in the future. OK, thank you.
Thank you, Anshul. That was great. I love to continue talking about that afterwards, and I'm sure you'll be around. Moving on, you referenced Sue-In's work on explainable AI, so come on up while I introduce you. Sue-In Lee is the Paul G. Allen Professor of Computer Science and Engineering at University of Washington, leading pioneering research in AI and ML across diverse biomedicine domains such as genomics, systems biology, healthcare, and beyond. Dr. Lee's lab is renowned for its expertise creating explainable AI, XAI methods, addressing a broad spectrum of challenges in biomedicine and beyond. Looking forward to hearing what you have to tell us.
Thank you so much. So my talk will be about the AI technology. side of the excitement we have here. I'm gonna talk about many projects that uses a AI technique. It's a subfield of AI called explainable AI for a wide variety of problems for biology and health. So before I start, I'd like to thank all the people who are behind the research I'm about to present. I work with nearly 10 or more than 10 PhD students pursuing computer science major. Some of them are actually from the MSTP program, meaning they are MD PhDs. And then we work on a wide variety of topics, including AI machine learning. The goal is to make machine learning models more interpretable, and then also basic biology, and then clinical medicine. Okay, so this talk will consist of 2 parts. So one of them I'm going to talk about several examples with the neurodegenerative disease studies as example, what explainable AI can do, why it is so important to make sense of biological data. In the second part of the talk, I'm going to talk about how explainable AI can help us find a new direction and pioneer new directions of research, including model auditing, for example, using various applications as examples. Okay. So let's say that you have a set of features from a patient, or not a patient yet, let's say a subject, and you have a well-trained machine learning model. You can predict various kinds of outcomes pretty accurately. For example, whether that person is going to have dementia in the next 3 years. Okay. This is an actual study we pursued recently. But the traditional machine learning paradigm cannot answer a very natural question of why a certain prediction was made. It's because with the large modern data sets, often complex models such as deep neural networks are going to outperform, achieve better accuracies, and then those are the models you will end up going with. But these are black boxes, and even experts struggle to interpret them. So many years ago, several years ago, we developed a method that's called SHAP, the idea being that you can estimate the importance of each feature for a particular prediction made on a particular patient or a particular sample. Okay. For single cell data set, it would be a cell. Okay. So that you can understand the relationships between input features and then output Y, so that you understand their relationship, regardless of how the model is complex. And this is a non-trivial computer science problem. I showed the first publication in December 2017, but we still publish a lot of papers in this space. To just give you idea of how it's actually used for our improving our understanding of the neurodegenerative diseases. So Alzheimer's disease, I don't have to emphasize how important to understand its cause, and then even early drivers, especially of this Alzheimer's, and then plaques and tangles are the neuropathological hallmarks, and then A beta and tau are the basic components of it. And then these days we see lots of data sets, genomic or transcriptomics or epigenomic data sets from various AD cohorts. So let's first see, I'm going to show you a first example of how explainable AI can be integrated with this kind of study. So my student, Nikasia Bebe Wang, a few years ago, she collected all this, the RNA-seq data. At that point, it was bulk RNA-seq data from various AD cohorts. So these cohorts has both of AD and then control brain tissues, all post-mortem. And then we modeled this complex relationship with neuropathological phenotypes using complex models. So here, the purpose is not to predict. These are all post-mortem tissues, and we want to understand the relationship between X's and then Y more clearly. We use the deep neural network, and then it comes with a couple of advantages, one being that the phenotypes, the neuropathological phenotypes, do not need to be perfectly matching across cohorts. So for some cohorts can miss some of these neuropathological phenotypes. You can still train a robust neural network model. And then also you can capture complex nonlinear relationship between gene expression levels and then these phenotypes. So the goal will be that you want to identify the drivers, the potential drivers of Alzheimer's, but it comes with this interpretability challenges. So I'm going to just summarize how explainable AI benefit this kind of a study. First is that since we were able to train the neural network model, powerful neural network model using multiple cohorts, we learned a pretty good model that can successfully recapitulate the relationship between gene expression levels and the neuropathological phenotypes, even in different species, and then different human tissues, like blood tissues. And then also using XAI technology, we can identify the genes that are important, that contribute to the AD neuropathologies, which led to previously unknown relationship between immune response genes and then AD neuropathologies, with other strong biological implications. That's the paper that I listed there, Nature Communications. In the next more recent paper, we also used the same idea, similar idea, to the problem of predicting drug synergy in cancer patients, acute myeloid leukemia using gene expression levels. So our approach also led to strong biological implications. So after that project, we really thought about how best, the better way to use explainable AI for this sort of genomic or transcriptomic research. So obviously, individual genes are not as interpretable as functional units, such as pathways or regulatory networks. And then plus, unsupervised modeling can sometimes be helpful. The idea is that you project your high-dimensional gene expression data into lower-dimensional embedding space so that you can reconstruct the data. It's data compression technique, crucial step in any kind of a single-cell data or transcriptomic data analysis. So what XAI can do is to identify the genes that can explain a large portion of the gene expression variation across the data set. And then you can interpret that as some sort of drivers of the expression variations of the genes. So combining these two ideas, we came up with an interpretable neural network architecture that has pathways in the middle layer. So in the compression pattern will be that you will know the activity level of each pathways based on the pathway databases. And then you have a sparse connection between genes and pathways so that it's aligned with your prior knowledge. So, and what that can tell you is that within the data set, you will know which pathways are important in explaining the expression variation across subjects, across the samples. And then you can also attribute back to the input genes to identify potential drivers within that pathway. So we now applied this approach to the extended bulk RNA-seq data sets from AD study cohorts. Obviously, we don't have to worry at all about the phenotype neuropathology availability, so we were able to gather more samples. And then my MD PhD student, Joe Janizak, applied this model to the AD data sets. And then we identified oxidative phosphorylation, or mitochondrial complex 1 as a potential important pathway within this AD case control study data set. Transcriptomics data set. And a really cool part is that we were fortunate to work with Dr. Matt Kabolin at UW that time, UW Pathology, and then we did in vivo validation with using the transgenic C11. So these are the worms that have Alzheimer's, and then we knock down the gene, and then each gene in this pathway, and there was a consistent increase in the lifespan of these worms with Alzheimer's. So that indicates, it can be a promising pharmacological avenue. There is actually a drug that's known to suppress complex 1, which is, one of them is capsaicin. Okay, quickly moving to the next project, and we noticed that many single-cell data sets are produced, and single-cell ATAC-seq or single-cell RNA-seq data sets from these AD cohort studies. And then one natural question we use when we perform single-cell experiments is the contrastive reasoning. So where to investigate the differences in the cellular state between background cells and those under specific treatment. It can be disease, it can be some drug treatment or genome editing. So we came up with another interpretable neural network that separates the latent space into shared variables that matter to all cells, and then also salient variables that can capture the expression variation within only target samples. And it's a recent paper in the Nature Methods, and you can find it in my publication, in my homepage at the top. So And then when we applied this method to cancer cells treated versus control, we see in the salient space all the cells behave differently depending on whether it corresponds to TP53 mutational status. And then you see that only because all the nuisance cell type specific signals are absorbed in this shared space. You don't see this otherwise, because it's such a dominant biological variation, the cell type specificity. So, and then now we are applying. With my collaborators, we are now pursuing research in terms of what AD, what different expression signatures distinguish AD versus the control brains, and then even finer grained AD pathologies. Okay, so quickly moving to the second part of the talk, what new research you can do with explainable AI. So first is model auditing. So during this COVID time, like in 2020, and then we found that many published AI models that use chest X-ray data to identify COVID-19 rely on shortcuts rather than genuine pathology. And then we first identified that when we did experiment with internal data set. This is cross-validation setting. You split the training and the testing data within one data set people used as training data. And then we collected another second set of external data and then did external validation. And then the AUC difference is what you see. And then explainable AI highlighted what was the most important parts on the chest X-ray image in each of this decision systems. So for example, it was right in that many of them were right in that clear long basis is it's a basis to say it's COVID negative. But lateral remarks, a lot of cases we saw this lateral remarks on the X-ray was selected to be important, and also medical devices as well. So many of this kind of XAI-based analysis identify this kind of mistakes all these model made, and then it turned out that the training data set was created in the right way. So meaning that they combined negative labels from one data source, and a positive COVID is more recent, right? And then this and then newer data sets that has most of the labels being COVID-19. Right? Okay, so and then it was really interesting enough that we are pursuing another research. Another domain is that in dermatology area you see a lot of cell phone apps. You take a picture of the potential lesion, and then you want to know whether that's melanoma or not. We got similar results, not as bad as the COVID example, but some of them were really concerning. And then the technical challenge there is the saliency map. This XAI, plain XAI method I told you about is not going to work because in a skin image like this, it's going to just point to entire region. It just doesn't tell you what is. It doesn't really explain to you why that kind of a prediction was made. So now then we need something more than this. So we developed a method to modify the image minimally so that the classifier changes its decision from benign to malignant. And then you see all this, it is called blue-white veil. The center, you see this bluish area, right? So that's a usual indication of a malignant. Okay, so that tells you that, that's what this AI model is thinking. It's reasoning that way. Blue-white veil means malignant for this system. And then it's consistent with the clinicians, right? So we showed, a lot of results in that, that kind of result. And then also we developed another project, another method that can also automatically generate the concepts such as, blue-white whale. Based on the CLIM model, based on the language, vision language model. Both of the papers are now in revision for Nature Medicine. Okay, so I'm gonna skip due to the lack of time. And then we developed a method also to for cancer precision medicine. So the idea is that there are hundreds of individual drugs. There will be tens of thousands of pairs of drugs. And then in that case, explanations in terms of predicted efficacy or synergy is going to be really important. So a machine learning model you can think about there is that you have gene expression levels of a patient, and then drug combination features. They will tell you what target genes they are, and then also molecular structure. Lots of features there, right? And then you predict the drug synergy for all tens of thousands of drug pairs. And then now you can extract what features were important. And then from this project, we discovered some underlying principles in terms of how drug synergy works. And then it all pointed to stemness, how the differentiation level of the cancer, and it's usually connected to prognosis. More differentiated, better prognosis, and less differentiated is worse, right? So, and then that suggests that it can be used as axis for designing combination therapy. If you want to use drug A that's known to work well for the well-differentiated cancer, you want to use B that's the other way. All right, so I just showed you a tip of the ice of our research in terms of explainable AI applied to biology and health. And then I want to emphasize that this is still a very challenging problem from computer scientist perspective. There needs to be more research to be done. And then also, we had a really exciting set of biomedical research as well. You saw some of them, and then, spanning medicine and competition, cancer biology and precision medicine, and then also Alzheimer's disease therapeutic target discovery. So you can find more about, our publications. And I'm so sad that I don't have time to talk about, more set of this. All I can do is just show you, this short citations. Okay, thank you.
Thank you, Sue-Ann. We have a bunch of questions that have come in for all of you, and they seem to have just kind of naturally mixed themselves in order, so I might jump around if one of you gets too many questions in a row. But the top of the list right now, Sue-Ann, is we'll do last first. Fascinating talk. 2 questions here. One was weighing individual gene contribution would assume independence of genes. Is pathways a first step to dealing with that? And the second question is, are you adding phenotypic features to genomic ones?
So to answer the first question, this is actually an excellent question because explainable AI techniques, we assume that the features are relatively more independent than what the gene expression levels really are. Gene expression levels are highly correlated with each other. They are modules, right? And so, pathway-based analysis helps with that because typically, genes that are in the pathway, they tend to be correlated or anti-correlated with each other. So if we use this pathway, based architecture, the pathway level importance would be the first. We should, get a sense of what the important pathways are, and then go, progress to the next level, which is individual genes. Yes, and then the second question is, are you adding phenotypic features to genomic one? So in our current study, past and current study I just presented, we use the phenotypic features would be in the output side of the model, but depending on what the scientific questions are, you can definitely incorporate phenotypic features as input also. I mean, if you are thinking about, say, like multimodal foundation model, then you will just put everything as the features, right? So yeah, it depends on the scientific context.
Thanks. Anshul, you're up next. This one echoes back to some of the work we did together a few years ago about you can only learn if you have data. So the question is, given your impressive methodologies and the challenges you displayed on your last slide, what do you think about, I'll paraphrase a little bit, about the opportunities for novel findings? Where are they data dependent? Where are they rate limited by data, and in particular on public data sets, and challenges with getting the signals that you need to train the next level of models and the next set of insights?
Yeah, I think it's a really interesting question. I personally think that we are actually not data limited. We've been training large models on tons of public data that's freely available. Can you hear me? Okay. And the current models that we have, they're finding so many super exciting, intricate findings. There's infinite hypotheses these models are able to produce. I think the major problem right now is these models sit in kind of supplementary websites of papers. So one idea is to collate these models into unified model zoos, have APIs that allow you to extract information from them directly through interpretable, explainable AI techniques, embed these into public knowledge bases and knowledge graphs. Maybe you can build language model interfaces for them so that people with different backgrounds, different expertise can communicate with these models and go back and forth. I mean, these models should be powering biological discovery. They should not be secondary or coming after data sets and after you decide what you're going to pursue. They are hypothesis generation engines, and you can use them that way, as I showed you in several of my slides. So we need to build the infrastructure and the technology to enable, next time a biologist decides to do an experiment, they should not simply use their domain expertise and their prior knowledge. They should query the world's public data sets, models distilling them, extracting really exciting information, and having communication back and forth. So that's what I hope we will go in the future, and I hope NIH and other public sources can fund such efforts going forward.
Thank you, Anshul. Angie, next question's for you. Do you make modification calls for methylation and hydroxymethylation, in particular from nanopore R10 in the BAM crams?
Yes. So yeah, I left out a lot of details because there's just not enough time. So everything in phase 2 will have methylation data. So the original 1000 that we just released, the technology on PacBio didn't have capture of kinetic data, but now it does. So everything going forward, both PacBio and Nanopore will have methylation.
Great. Suan, back to you. You made a comment about no need to harmonize phenotypes across cohorts. I think in particular we're talking about Alzheimer's and some of the different ways symptoms were captured. Was it simply that the AI was robust to whether a variable was absent, or was it somehow resolving, merging, combining diverse phenotypic descriptions?
So it's about just how the neural network is trained. So let's say that you have this, we use the 680 neuropathological phenotypes, and once you assign, well, actually, there are multiple ways to measure, A beta, okay, or tau, and then let's say that you have a set of phenotypes you want to go with, and then for each phenotype, you will have this set of samples that have that phenotypes, right, set of cohorts, and then those are the samples you will be using. So it's just the nature of the deep learning, how the deep learning training is working. It's modularized in that, the connections, the weights between variables can be trained based on, what data, whether you have that variable existing. So it's more about, more fundamental property of the deep neural network training. Yeah, it's not like AI magic. Yeah, modularization of the neural network.
Thank you. Angie, are generating or planning to generate WTS in addition to WGS? Now, never, later?
Yes. I'm planning a lot of things. Another thing I didn't get into. So what we want to do is within this selected group of, this pilot group of 10 to 15,000 participants, is to go deep multi-omically. And we're excited to be starting doing some proteomics and RNA-seq in the next few weeks. And anticipate that data should be fully, completely generated by June.
And confirming that is a choice to deeply characterize the same subset of individuals on a
Correct.
bunch. Yeah,
And then metabolomics is next.
I think this next one is for you, Anshul.
Yeah.
How much does the variance influence have on regulation based on one reference for training?
Yeah, it's a very interesting question. So we had hypothesized that if you trained on personal genomes where you actually encoded the underlying, the genetics of the sample from which you got the data, the model would do substantially better. We actually, at least from our models, we do not see any significant improvements. But there's a plus side to that, which is we've actually done, I didn't get a chance to show it, but cross-population analysis. So you can train models in reference data from European individuals. So it's pretty difficult to get every cell type and cell state in thousands of individuals from every population that you can think of. Hopefully we can, but the current state of the art is that you can get lots of good reference data sets. We can train models in these on reference data sets from any reference population, and we actually see almost no degradation, unlike what you see with association studies like polygenic risk scores and GWAS and so forth, where linkage disequilibrium and other kinds of artifacts, cross-population artifacts, really result in sudden drop in performance as you switch populations. In our case, if we go from, for example, European ancestry reference samples to African subpopulations, we actually see zero degradation. And I think the reason is because the models are learning underlying causal biology versus looking at statistical associations. And in many cases, the underlying biology is consistent between populations, even though the genetic architecture and LD structure and so forth change. So in summary, it'll be great to train actually on pangenomes and things like that, but I don't think they're going to be some magic bullet to suddenly make everything work. The models are already working very well. They'll probably give you a small delta going forward.
Right. We are not going to get to all the questions here. I'm going to ask, I think, one more question for each of you, and then let people get to their break. And I'm going to ask some questions that are not quite on the screen. I'm not going to let you, Anshul, answer the bottom one there, which is I didn't know you brought your fan club. It's great. I agree. But so, Angie, I guess my question for you is, as you think about the data that's being released, in particular phase 2, which is a nice, what do you hope people will do with that data? What are you looking for as papers published a year after the data's out? Or what are some of your hopes for that?
Yeah, my vision is that with these additional data types, we will actually start to be able to characterize previously unseen variation, particularly in the underrepresented populations, and start to level the playing field a little bit with our reference data sets and the ACMG genes and all of these known functional genes and diseases, and we've already seen in our own data that there's an increase of VUSs in these populations, and let's start to characterize some of those and really start to chip away at that.
All right, thank you. And then maybe I'll ask the same question, both Anshul and then Sue-In. The new buzzy round of generative AI and the very large models using it, how much do you think that will influence the kinds of work you're doing? Is it just, different tool, different space? Is it fundamental? Is it, does it create a whole new set of explainability, a whole new set of, just first thoughts on, and maybe Anshul first and then Sue-In and then we'll wrap.
Okay, sure. Yeah, I mean, I think Sue-In will say the same thing, that I'm a big fan of all these new classes of models coming up. I worry a lot about, borrowing the strategies of how people work with these models from other domains, where explainability and understanding and reasoning and causality is not the primary objective, whereas in genomics, genetics, biology, that is pretty much the entire point of doing all of this. And so it's a different domain. And so I think the few of us who work in this sort of explainable AI space, especially focused on biology, I think it's very clear that as these methods continue improving, generative AI models and big LLMs and so forth, it is going to be even more important to continue building powerful interpretation tools so you can debug models, find when they pick up weird artifacts, when they're learning assay biases, and help extract the biology out of them. So I'm very optimistic. At the same time, there's huge roadblocks to making them really work for, I think, these applications that people in this room potentially care about.
I'm also hugely excited about generative AI. It's now changing the landscape, not only science but also society too. So yeah, my lab is pursuing research in terms of just how to interpret this generative AI model. So the one example is that when it's trained with like lots and lots of data and then generated something, text, or it can be data, and then you want to know why that kind of output. I mean, which training samples were important? Well, to ensure that training samples are fair in terms of it, it's across all diverse group, of the sources, and then so I think generative AI is making the field of explainable AI even more active, and then we are really excited to develop new XAI technologies for this generative AI. And then one of the examples I showed you is it actually the other way. I really like this feedback loop. Generative AI helping the explainable AI, because collecting the data, medical data or biological data, it takes a lot of time and a lot of money. And then let's say, data were generated, and then one use case, really exciting use case I can think of is to stress test machine learning model. So one example I showed you, the skin, skin image, you change a certain aspect of the skin image. If you are successful, you can see the reasoning process of your machine learning model, right? How blue-white whale is, considered when they, make a prediction in terms of melanoma or not, right? So I really am excited to see this like feedback loop, XAI helping Gen AI helping XAI. So I'm overall really excited. But really, it's related to one question that appeared there. The ethical consideration. We really need to make sure that to use XAI to really understand what generative AI is doing. Especially in terms of the training samples.
Great. Thank you all very much. Unanswered questions will be in the Slack, and time for our break. Thanks, everyone.
been finding the time to be bored out of my mind. I've been walking for days. I don't have the time to play your kind of parlay. So won't you come home soon? Won't you come home soon?
Hello? Hello, GA4GH. Hello, hello. We are ready to start. We're ready to start the next session. We have another set of great speakers. All right. Thank you, everybody. Come on in. Doors are closing. Take your seats. We have another session that should be exciting about some of the connections between what we do here on a pure research academic governmental side and how to get government and academia and involve industry with that. Different sessions will be talking about that, different speakers talking about that from different angles. First off is going to be Melissa. So Melissa, if you want to come on up. Melissa Konopko should not have walked away from the mic as I was talking. Yeah, Melissa is a scientific product manager at Elixir, working within the Human Genomic and Translational Data team. Her primary focus is on creating and harmonizing tools that guide countries along an international interoperable pathway, as we heard about yesterday, to advance their genomic and related data infrastructures. Melissa led the development of the Million Plus Genomes Framework and the Elixir Human Genomic Data Maturation Model. These key initiatives are being adapted for use in GDI, the European Genomic Data Infrastructure Project. Her educational background includes a PhD in neuroscience and an MBA from the University of Maryland. Melissa, all yours.
All right, so I am here representing the great work of Despina, who is our Industry and Innovation Officer. I'm going to try and do my very best to show off the great stuff that she does, but try and go easy on me on the questions, because I probably don't know the answer. All right, so I'm going to first start talking a little bit about why industry matters to us. And then I will talk about Elixir, because I know you guys have heard a lot about it. You keep seeing our little symbol showing up on various slides. And I was talking about the Genomic Data Infrastructure Project yesterday. But it's just a small part of what we do. And I will then bring those two pieces together and talk about what our industry engagement strategy is. And then finally bring it over to focus in the area of genomics and other human data, since that is going to be the thing that is most interesting to everyone here. So this is a preaching to the choir slide. We know open data drives innovation, in particular being able to join various kinds of data together helps both companies as well as academic researchers make progress. When we spoke with various SMEs about open data, they found that combining both open data as well as their proprietary data through their own business innovations brought them to create the outputs that people at Elixir and across GA4GH really care about, including diagnostic tests and drugs, medical devices, and technology solutions. Further in this research, we spoke with these SMEs, and they said that 76% of them found that without open data, they wouldn't be able to provide the kinds of products that they have created. And this graph here shows over time the number of patent applications that mention various Elixir resources. So in the past 5 years, there's been around 1,500 per year that come out that required Elixir resources in order to accomplish what they have. So I'm going to give everyone a little bit more background on what Elixir is. And I think it took me a while even working there to even understand what Elixir is, because I think everyone sees a little part of it. It's that classic story with the elephant of you only see one piece, and it becomes a lot more complicated. So I'd like to give you a little understanding so you can understand why we engage with industry. So we are an intergovernmental organization to coordinate bioinformatics resources across Europe to form a single infrastructure. So this includes databases, training, software tools, standards, compute resources, and scientific and technical experts. And so that's when we're coordinating a lot of these large projects that I mentioned before, like GDI, so bringing people together in order to accomplish these big things. We have 23 member states across Europe, plus Israel. And each of these member states is comprised of several organizations. And so we have 245 institutes and universities that make up Elixir. And we are coordinated at the hub outside of Cambridge, UK. So you might wonder why an intergovernmental organization that's kind of on the nonprofit-y, more academic side would care about fostering innovation. So we are funded by European level and country level sources, and a lot of countries want to see SMEs and innovation to thrive within their countries. I want to put a little asterisk here because since we are trying to represent such a broad number of countries, every country has a different view on the way that industry should be connecting with something like sensitive human data in particular. And so we have to represent some of the more conservative viewpoints when we do engage with industry. So while we do want to see industry thrive, we need to be very, very cautious about the way we do it. We also can go to Central European sources as well as to countries, and since we have interactions with so many different countries and organizations, and we can act as a central body of knowledge to help shape governmental priorities. In order to create this infrastructure, we need to make sure it's actually representing the needs of bioinformatics data users across Europe, and so we have 15 communities that cover many, many things. So a lot of people here probably know about the human data ones, the federated human data, human copy number variation, and rare disease. But we also cover things like plant sciences and marine metagenomics and research, data management, and food and nutrition. So we do cover a really broad range of data types at Elixir. So I'm going to talk to you now about understanding what Elixir is and what we do, and why we care about industry. I'm going to talk about how we engage. So we have these EU projects. You've heard me talk yesterday about the Genomic Data Infrastructure Project, but we've had several that engage more directly with industry, such as FAIR Plus was a big one, also in terms of COVID infectious disease, and with genomic data standards. We also engage through Elixir nodes and communities, which help give us more of this national perspective. And we have some more strategic collaborations with industry. A group here that you guys probably know more about would be the Pistoia Alliance. I'm surprised to Alex Kennitz. His face probably didn't expect to be showing up in this here, but he does this really great presentation on the Elixir GA4GH cloud, and that was given as a webinar for the Pistoia Alliance. So with these three pillars, we have different ways that we engage here with industry. So we supply services. I do recommend, if you're interested in all in this, get onto the slides and click all of these links. We supply services, including collections of case studies and business models. And we demonstrate impact with nodes good practices. We engage directly with industry through events, and I'm going to talk through a bunch of those later, as well as newsletters and social media. And we have a knowledge exchange scheme. One of the main industry events that we've had in the past, where we've had some successful collaboration, the Elixir Biohackathon 2023, where Microsoft worked with us on the Biohack Cloud using GA4GH standards. We also do some capacity building and matchmaking, including listings on a vacancy page, but we also have an industry advisory committee, and it's open to new members. So if you are a company representative and are interested in working on this, I recommend scanning this QR code now, or getting onto the slides, as I mentioned before. Since I see people scanning the QR code, I'm going to give it a second before I switch the slides. I'm glad to see this. All right, fantastic. And then, since GDI is one of the big things that we're doing, I'd like to spend a little bit of time talking about how organizations can engage there. So you saw this yesterday, but I just wanted to point out that the large timeline and scale of the Genomic Data Infrastructure Project. And we have these three pillars. And so we're focusing on making it last, making it work, and making it useful. So in the sustainability pillar, or making it last, that group is working on business models, as well as creating flexibility for different countries. So if a country wants to use Google Cloud or Amazon Web Services, they have the ability to do that based on the flexibility and the structure we're creating there. If they want to be able to engage with different solutions, they can do that. And so this gives the space and the flexibility for different countries to find their own solutions. Pillar 2 is going to be working on creating the main technical infrastructure, but it's also going to be a place where we're going to find some of these key gaps that need to be filled. And this is a great place for SMEs to step in and start to create some innovative solutions. We've already been working with REMS as an example there. Pillar 3 is the making it useful, or the use case focused pillar that's working with applications and innovative solutions. And so we are going to be finding a lot of open areas here for innovation. And it's a great way for people who are outside of the organization and SMEs and in general to be finding solutions there and to be looking not just to GDI, but also to the countries to be able to offer solutions. This is a project that Despina, along with Giselle Carey, have been working on creating this interactive map of the industry landscape in genomics. And so we went to various SMEs and asked them about the kinds of tools and services they have across genomics to benefit all. And it's an interactive map where you can mouse over each of these companies and see the solutions that they have. For example, we have some work from GenXT, and we have those guys in the room here, so they can talk to you about some of the things they have there. It is open for more companies to get involved if you want to show what kind of solutions you have to the landscape here that is available. And one of the things that we found when talking to various companies is that they said one of the main issues they have is that there's a need for adoption for data interoperability and using data standards in order to be engaging with these various resources. Just quickly talking about the industry engagement opportunities we have coming up in November in London. There is a Elixir Bioinformatics Industry Forum event, and it's going to be focusing on trusted research environments. So if that's something that's interesting to you, the registration is open for that. We have the GDI Virtual Stakeholder Forum, so there's the ability for industry members to engage directly with GDI there. And Biotech X is coming up next month as well. So all of this can be found on the Elixir website under the events section. So if you go to elixir-europe.org, you can easily find all of these if you didn't manage to get the photos that you're hoping for there. And that is really it. I just want to say, you can, if you're interested in following along with some of this, you can subscribe to the Elixir Industry newsletter. If you have any questions that I am unable to answer, then definitely contact Despina and she can help you out with that. Thank you very much.
Thank you, Melissa. Great to see one of your other hats on display here. All fitting together well, which I think is the point of industry engagement with the technical and scientific sides and pulling it all together. Our next pair of speakers, Bartha and Shane, if you, there you are, you want to come on up. Are going to talk to us about genomic data sharing by health technology industries and talk to us about some of the things to consider. So we'll do brief intros here. Shane Chase is the Global Head of Privacy, Ethics, and Policy Governance at Illumina, role that she's held since 2019. She holds a Juris Doctor from the University of San Diego, Master's from l'Institut Politique de Paris, and a bachelor's degree from UCLA. Bartha, I feel funny introducing you to this group. I think everyone knows you, but I will read your introduction. Professor Bartha Marie Knoppers is a full professor and the Canada Research Chair in Law and Medicine and Director of the Center of Genomics and Policy of the Faculty of Medicine at McGill University.
Thank you, David, and hello, everyone. It's a real pleasure to be here. with Shane Chase from Illumina, and I think you're going to hear about a very exciting initiative, which I have to say actually was industry who came to the Center for Genomics and Policy and asked us to begin working with them in a way to recognize the partnerships that have been in GA4GH, as we saw yesterday, since its beginnings. So we decided that we wanted to prepare industry-specific, if you like, norms, taking the ethics and rules work already very much present in GA4GH and translated into more industry-specific ethics and points to consider that could be used now and in the future. So why did we do this? Well, we heard the word partnerships, and we know that the translational route is very important, and we know that the framework that founded GA4GH needs to cover all the sectors and all the partners that are involved. And so this was quite an obvious gap, so I want to thank Illumina for having recognized it and saying, let's do something about this. And we know, particularly in the policy and ethics field, that there's always this hesitancy about commercialization, and are the participants from whom the data and samples came really recognized and treated in an equitable fashion. So that was one of our main concerns, how to do responsible international data sharing in a rapidly changing policy landscape. So these are the points to consider, which you can find at the DOI. You see at the bottom of the screen. You also note that health technologies industries, I think we might have invented the HTI acronym, I'm not sure, includes pharmaceutics, sequencing, clinical genomics, direct-to-consumer testing, digital health management, cloud computing, advanced analytics and AI, as we just saw this morning, wow, and digital health and engagement platforms. So that's quite a typology, quite a challenge. So how then do we prepare points to consider that doesn't get confused with the code of conduct and the GDPR in Europe, is not really a best practices which we should leave to particular professional norms by sector, and yet cross all these diverse fields. So we wanted to avoid legal jargon, and we wanted to make sure it could be used, and each of those points could be translated into more specific, maybe best practices, or maybe a code of conduct by particular industries involved. And look at this. These are the partners. It's kind of scary, I have to tell you, at least at the beginning it was, that came in on this project. And over 2 years, we worked together all the way from direct to consumer, through the cloud, to pharma, to AI, to digital, to Genomics England, and patients, the clinical setting. And by bringing together, I think, this very representative and diverse group, we hope to have prepared points to consider that can be translated into more specific codes or best practices. We use the core elements from the framework of the Global Alliance as a starting point for our work. And most of all, we wanted to, how do I put it, use the unique human rights approach that we have in the Global Alliance, which is to get past, common law versus civil law, ethics versus this and that, and all the binary kind of division language that challenges us to come together as a global community and use a human rights approach. The right of everyone to benefit from science and its advances, and the right of everyone who invents and creates to be recognized and receive attribution. A very important and universal human right. So, in conclusion to my little introduction, because Shane will go through the points to consider, we had to constantly be aware of this shifting international environment, especially as concerns data sharing. And just recently at the bottom of this slide, you can see, I think it was a month ago, or maybe 2 max, the US and the EU finally came to a privacy framework for commercial transfers. So you have to keep in mind how then, where lie the areas of consensus, and then build on it in this particular environment. So I'll close with this. You can see people think policy grows on trees. It doesn't. And you don't dream it up at night either, at least try not to. So we did our research at the Center for Genomics and Policy. We created 8 thematic briefs that sort of began our discussions on the 8 points to consider, which Shane will show you. And the points to consider themselves, each theme has points, and the whole document has explanatory notes and the thematic briefs, which constitute like international context for the themes. So with that short, hopefully, introduction, I'll pass it to Shane.
Thank you, Bartha, and thank you to everyone here for the opportunity to speak to this group. It's been my first time at this meeting, and it's been such a pleasure to be here and to meet so many of you. Okay, I know how to work the clicker. So as Bartha described, the first phase of this initiative was to do this landscape analysis to try and arrive at some consensus on what are the key issues that we want to focus on for this initiative. So we arrived at these 8 categories that were the focus of the second phase of the initiative. And CGP developed a series of thematic briefs on these topics to try and scope them. I say try because these are really interconnected concepts, and there's a fair amount of overlap, but they did, as always, really excellent work. Scoping each of these concepts. It also provided a common basis for conversation in these bi-monthly meetings that we had with the industry core group to allow for the industry to have again, common basis for conversation, but also an ability to collect feedback internally that then they brought to these meetings. And then finally, it helped us to drive towards arriving at what became our 8 points to consider. So again, the purpose of this was to try to bring together a group of diverse companies who are all working in this highly complex international space and having to grapple with very differing privacy rules and regulations across the globe, and see if we could arrive at a set of common priorities and approaches for enabling responsible sharing of genomic data. The idea was that this would help to support stronger and consistent data governance practices and be part of a broader effort to support greater transparency and public understanding of genomic data sharing aims and practices. So the first category here is, and maybe before I go into more detail, I'll just pause to say, what you see here is a very high-level summary of the points to consider that we created. I don't want you to leave thinking that after several years of work, we only arrived at 2 bullets per category. That's not the case. There's actually a lot more in there if you pull it up online. So with respect to consent, this is very often a necessary legal requirement that has to be met when collecting or sharing data. However, so we cover that, of course, in what we discuss in the points to consider, but we also discuss the fact that particularly in the genomics context, consent is often unnecessary but not sufficient because as technology gets more complicated, it's really impossible to arrive at truly informed consent. And so we also talked about how opportunities to be able to support consent through other sorts of safeguards, things like oversight committees, making sure, striving towards transparency, and the right to be able to withdraw consent. I will also note here that there's clearly a lot of GA4GH guidance on this point that we thought was highly important. And so one of the things we hope will be useful coming out of this initiative is that CGP helped us to look through the available GA4GH guidance and pull together a list of some of the most relevant GA4GH guidance for the areas that we covered in the points to consider, and that is included in the final document as Table 1. And so our hope is that that will help to promote awareness of this relevant guidance for industry and drive greater attention to it. The second category is transparency. Again, this is a concept that everyone's going to be familiar with, and it was really kind of one of the driving forces of this entire initiative. But the basic idea here that we talk about is the need to be able to describe the scope of data collection and use, as well as any potential storage and sharing of data. This is an area that's getting increasingly complicated to be able to do to achieve this kind of explanation in a way that is understandable to most people, particularly now that AI has kind of burst onto the scene. So, adding in here the ability to be able to describe the use of AI in health products and services is something that is just adding another layer of complexity to this issue. Responsible use is the third category. Here, the main idea was finding a balance between two imperatives. On the one hand, the need to comply with legal and ethical requirements for data sharing, and on the other hand, maximizing the scientific, the commercial, and the societal value of genomic and health data. And so, again, trying to, with kind of those guideposts in mind, coming up with some agreed data governance techniques to be able to push towards that. Identifiability is the fourth category. All of the company representatives that were part of this conversation really agreed that identifiability is kind of this key factor that so much turns around, because the level of identifiability is the key determinant of data's privacy risk profile and regulatory status. And it's really kind of an evolving issue in genomics. As technology evolves, what is identifiable? And also the regulatory context on this point differs enormously. So in the EU, many of you probably know that genomic data is special category data, so the most highly regulated category of data, whereas in the United States, a whole genome sequence is not considered to be personal health information unless it is attached to one of the 18 HIPAA identifiers. So just completely different regulatory contexts. And so we discussed practical approaches to manage privacy risk in this environment, while recognizing that there are trade-offs between protecting privacy and preserving the utility, quality, and representativeness of data. I see that I am almost out of time, so I'm going to go a little bit more quickly. The next two categories are privacy and data protection, and data security and quality. We did our best to synthesize, the key ideas here that translate globally and that are most relevant for this context. I think, maybe a couple points to call out that I think are of interest is that I think AI has raised, and the way that it really, again, kind of burst onto the scene about halfway through our work, raised some new and interesting questions in this context. So, for example, the hunger of AI and the need for AI to have these large representative data sets to be able to produce accurate and high quality algorithms is in direct tension with one of the core data privacy principles of data minimization. And so, what is the approach to balance those imperatives? The next topic is academic partnerships. We all recognize that partnerships between and among HTI and academic researchers are imperative to being able to translate scientific findings into health applications. And so, but we don't want, our objective was to try and identify what are some key governance strategy or key governance elements that we could pull together so that there's not a need to kind of recreate the wheel every time we put together these partnerships. And then with respect to community engagement, I know this group is in agreement with us that this is absolutely essential and that patients and communities have to be engaged in the development and governance of research. So not only do we need to build on existing effective mechanisms to do that, but also be open to creating new ways to promote community education, awareness, and understanding of data sharing aims and practices in a way that can support more informed and equitable public access to the benefits of innovation. So I'll move on to acknowledgements. You'll see all the names and titles and companies that were involved as the industry core group members, but most importantly, I want to thank Bartha and Jan and the team from McGill who were just such incredible partners, and it was really such a privilege to get to work with them. And finally, with 2 seconds left, here is where you can find the points to consider. It's also on the CGP website, but if you pull up this website, you'll also be able to go to it directly. Thank you all so much for your attention.
Thank you, Shane and Bartha. Our next speaker from Singapore is on her way to the podium. I'll start introducing her now. Dr. Seow Shih Wee is a Director of Corporate Services at PRECISE, the Central Program Management Office, coordinating and implementing Phase 2 of Singapore's National Precision Medicine Program. As Director of Corporate Services, Dr. Seow is responsible for coordinating daily operations across various departments in PRECISE and monitoring ongoing research activities and progress. Dr. Seow also oversees PRECISE public communications, staffing, and budget management matters, and liaising with various government agencies to support PRECISE operations.
Thank you. Thank you very much. I'm Shih Wee. Now I know why they keep saying press the green button. Okay, I will, yeah. I'm Shih Wee from Singapore. Thank you for giving me the opportunity to share Singapore's experiences. I must inform that I have hung up my pipettes many years ago. I have been doing scientific and research administration for the last 10 years. So any technical questions, even the simple ones, I will have to refer you to my colleagues, Maxime and Nicolas, who are sitting in the audience. So this is the brief outline of my talk. I will start with an introduction to sunny island, Singapore. Then I will describe Singapore's research funding landscape so as to provide context as to why the National Precision Medicine Program places a strong emphasis on enterprise and economic goals. Then I will conclude my presentation with 3 examples of how the Singapore Precision Medicine Program has powered the local ecosystem. Singapore is an island city-state located at the tip of the Malay Peninsula. We are a small country in Southeast Asia region. East to west, we just stretch 31 miles, and north to south, 17 miles. So if you're a marathon runner, you have no problem running across Singapore. We are located 1 degree north of the equator, so tropical weather and practically sunny all year round. We rank highly on various global indices, and we are one of the longest living human beings on the planet. Singapore is also where the Trump-Kim summit was held. It is also, I didn't expect to draw so much laughter. It's a fact. It is also the basis on which the novel and movie Crazy Rich Asians were based. And also we are home to the first ever sort of night race, Formula 1 night race. We just hosted the 15th installation over the weekend that just passed. I share the island I call home with 5.64 million other residents, 3.55 million of whom are Singaporean citizens like myself. There are 3 major ethnic groups in Singapore, the Chinese, the Malay, and the Indians. And the composition of this ethnic mix has remained reasonably stable for the last 10 years. Farm bio and medtech industries are significant contributors to Singapore's GDP and employment. We manufacture 6 of the top 10 drugs by global revenue in Singapore, and we are also the Asia HQ for over 60 multinational medtech companies. The reason for the success of the farm bio and medtech industries in Singapore is partly due to the steady and sustained investment from the government in research and development so as to create a knowledge-based economy to attract foreign investments. Every year, the government commits about 1% of our GDP to research activities, including building research infrastructure, funding PhD scholarships, and so on. And a key emphasis of the research funding from the government is translational research and also commercialization. Precision medicine was one of the topics or domains that was identified that can help to drive Singapore's research, innovation, and enterprise agenda. And it is for this reason that the government funded the Singapore National Precision Medicine Program. It is a 10-year program starting in 2017. And at the end of the program, we expect to have sequenced 1 million, up to 1 million Singaporeans. That's almost 30% of the Singaporean population. It is a whole of government and whole of Singapore healthcare and research ecosystem endeavor. You can see all our partners listed at the bottom there. It's been a very exciting journey. So NPM Phase 1 ended in 2021 very successfully. The team sequenced over 10,000 healthy Singaporeans. And despite being a modest sample size of just 10,000, there was active and healthy interest from the various industry sectors that you see on the bottom right, which is unfortunately not presenting well. So if you see Singapore, you would have caught a glimpse of Asia, at least genomically. We found that in our 10,000 genomes, the Singaporean Chinese genome, as represented by the red spots, closely match or map onto the East Asian genomes. The Singaporean Indian genomes, as marked by the purple dots, closely match that of the South Asian genomes. And this did not come as a surprise to us because the early settlers of Singapore came from the southern parts of China and also the southern parts of India. What did come as a surprise was that the Singaporean Malay genome, which is represented by the green dots, did not map onto any of the publicly available genomes that were available at that time. And this underscores or points to the general and global lack of Asian underrepresentation in public genome databases. This is also probably why over 40% of the variants that we found in our SG10K cohorts are actually novel and not present in the two major public databases. We need to address this gap. It is important because even in our small 10,000 cohort, we were able to find significant ethnic differences between the three groups. For example, in the Malay ethnic group, they have a higher likelihood to develop or to bear the BRCA1-2 gene mutation, and so they are more likely to develop hereditary breast and ovarian cancer in their lifetime. In the Chinese, they're more likely to develop familial hypercholesterolemia, it's a big word. And in the Indians, they bear the variant to MYBPC3, which predisposes them to hypertrophic cardiomyopathy. We are now in the middle of phase 2, where we look to enroll 100,000 Singaporeans. And all of them, each of them will contribute out over 1,000 phenotype parameters. And if delivered successfully at the end of the program, we should be Asia's largest and most deeply phenotyped cohort. And the depth and also the quality of the data would be able to match that of the UK Biobank and also all of us in the US here. Apart from research pillars, we also have health and innovation pillar. As you know, healthcare systems are generally very large and inert to innovation and also changes. So rather than going in big and upsetting everyone, we funded 5 clinical implementation pilots across 5 different disease areas that were relevant to Singapore. So the pilots allowed the doctors to run some hypotheses and also to try and do little tweaks to their clinical workflows, such as introducing genetic testing much earlier on in their workflow, and to ascertain and see how it impacts patient healthcare outcomes. A key component of these clinical implementation pilots are also health economics analysis. The reason is so that we can collect the data points to convince or to assure that the Singapore government and policymakers that although precision medicine may cost a lot at the beginning, but in the long run, the cost benefits and also the socioeconomic benefits will bring a lot more benefits to the Singapore population. The third pillar of the NPM is the enterprise pillar. There is a lot of interest in the Singapore Precision Medicine Program, primarily because of our Asian diversity, the deep phenotypes that we are collecting, and it's a longitudinal cohort. Also, the fact that it's a whole of Singapore and whole of government effort signifies our commitment to making this project work. So to capitalize on this interest, the team devised a 3-stage economic strategy to capture the economic value of the NPM. There are 3 stages. They amplify and augment each other with the end outcome of stage 3 to bring and return the investments and also the data resources that have been created to avail them to the local ecosystem and enable them to do the R&D and also commercialization. In phase 1, stage 1, we form important partnership with technology companies such as Illumina to create quality data sets so that we can attract pharma companies to co-invest in our program for data enhancements and other collaborative research. We know that genetically validated drug targets are more likely to lead to approved drugs. And that's why we see that pharma companies are very interested and very keen to tap on these national resources that are usually publicly funded and reasonably well-resourced. We have seen the success of the UK Biobank and FinGen in collecting and attracting repeated rounds of investments from pharma companies. So we are inspired by that and we would like to emulate that. I'm pleased to mention that as of August last year, we have received 4 in principle commitment from 4 pharma companies, and negotiations are well underway to create a master research collaboration agreement. And I hope that when we get invited to speak again next year, we will be able to share our experiences in setting up this pharma pre-competitive consortium. So the NPM has provided many business opportunities to the local ecosystem. I'll give you 3 examples. The first being our partnership with Illumina. Illumina is a sequencing technology company. They do not offer sequencing services, as far as I understand, or at least in the Singapore context. They do not offer sequencing services. And so they outsource the sequencing service component to a local company called Novogene AIT. And because of the guaranteed base load of 100,000 genomes from our program, immediately Novogene AIT could register an increase in their revenue. And also it allowed them to scale and expand their operations, creating good jobs for Singaporeans. And lastly, because of the scaled up operations and increased revenue, they were able to expand into markets well beyond Singapore. The second example comes from our clinical implementation pilots. 3 of the 5 clinical implementation pilots required clinical-grade genomic sequencing, which was not widely available in Singapore. Life Strands bidded for the project, and they are now in discussions or have formed a partnership with Embry Genetics based in the US to do a technology transfer so that Life Strands can set up an end-to-end clinical sequencing service from sequencing to generating a clinical validation report. So we're very excited about this. My last example demonstrates the translational and also the commercialization aspects of the NPM. The snapshot, the UMAP you saw just now, what I did not mention just now was that 26% of our healthy SG10K participants were found to have a carrier risk of severe recessive disorders. And yet, if we screen these individuals using commercially available panels that are developed based on the Caucasian European populations, they would not have been picked up. So this does not work for our population. So to circumvent this and to get around this, the doctors and the researchers have come together and are productizing an Asian carrier screening panel with the National Diagnostics Development Hub so that we can use the Asian screening panel as designed for Singaporeans and Asians for support to do preconception and carrier screening, and also to support treatment decisions. So this would be one of the first made in Singapore export-ready panels to service the Asian population. So I have given the 3 examples, and the project is very well underway, and we will have many more examples to share. So please invite us again next year. Thank you very much.
Thank you. It was actually great to hear the different perspectives on both why we should step on the gas and the benefits, and why we should have brakes handy to avoid the downsides and some of the balances. And so I like the panel covering all sides. If we could switch to the questions, we have a few questions in here. And we'll just go through these in order. And again, if somebody gets overloaded, I may jump around, but it looks like it's pretty well mixed. So Melissa, starting with you. You had mentioned in your presentation talking about a space, for example, an SME in Pillar 2. What's the underlying business model for an SME? What's the exchange of value? Are they selling a service, selling IP software? Who's the customer?
So I have to be very careful about the way that I answer this question. So I would say that we can, within Pillar 2 in GDI, show where the gaps are, and if there is a solution that either already exists or is close to filling the need that we have, that they can propose to either the project itself or to the individual countries that there is a solution potentially available. We have to be very careful within European Commission funded projects to not latch on too strongly to any specific solution that is provided by a company. So I would say if you're interested in this, we mentioned there's the industry-focused stakeholder forum for GDI. I would definitely recommend participating in that and expressing some of the views that you have and finding the best ways to engage with the project there.
Thank you. I think the care with which you answered that is part of the whole point of this conversation, is saying that we all recognize there's real value and there are real sharp edges, and that it's worth doing carefully, and that both parts matter.
We can't complete something like a European-wide and beyond infrastructure for bioinformatics without industry, and the whole purpose in creating this is to be able to support healthcare and to support drug development, and so we need to be able to engage with industry. We just need to make sure it's done in the most ethical way, in the way that keeps not only the governments happy, but keeps the citizens happy, because without citizen trust, we've got nothing.
Yeah. Dr. Seow, what are the plans for international data sharing from the Singapore cohort? Data access policies, who can request? Can you request now? What can you do with the data, et cetera?
Yes, so definitely interested to collaborate so that we can share the data set. In fact, our International Advisory Committee has said that the Singaporean population size, the Singaporean cohort of 100,000 is too small. So we should collaborate and validate our findings with other cohorts. Certainly keen. What's available immediately now is the SG10K cohort. I think you can go on and request for data access. So all access goes through a data access committee, which will assess and evaluate the ethics and also alignment to the overall goals of the NPM program. Phase 2, we are beginning to collect the data set and to tidy them up. It will not be ready till maybe the end of 2025. So yeah, please keep your eyes glued.
Thank you.
Thank you.
I think the next question is for you also. In precise Pillar 1, the population cohorts, do you incentivize participation? So what's the quote-unquote contract with the individuals, with the Singaporeans who are taking part? What's the exchange of value there?
Exchange of value is a $50 shopping voucher at, no, really, at one of the big grocery stores chain. We found that Singaporeans are generally very altruistic, very keen to participate. There is overwhelming response for the phase 2. As I understand, there is like a 1 year wait to get the assessments done. Let me see what else. We don't return beyond the basics of healthcare information, such as your blood glucose, your blood pressure. We certainly don't return genomic information at this juncture because it is mostly research. We do have plans to return incidental findings, and we are collaborating with one of the hospitals to do that. Yeah, that's all I have to share.
Okay. Yeah, I mean, I think that one is interesting just because of the number of different answers that the large programs have taken to that set of questions. With, I think of UK Biobank slightly oversimplifying at one extreme of, nope, it's research, you know, and we're not going to, you know, that's the deal, and all of us at another extreme of the whole point is return of value and information. Sounds like you're taking a balance somewhere in the middle there.
Yes.
Yeah. Bartha and Shane, indigenous data sovereignty. Has indigenous data sovereignty and benefit sharing and data use been touched on in the governance conversations that you've had?
Yes, it has. Believe me that we had excellent conversations about this, especially, of course, coming from Canada. We're very sensitive to the rights and health needs of Indigenous populations. Shane didn't have time to read all the elements under community engagement. The points to consider are more elaborate. And one of them, in addition to what she brought up on the screen, was different forms of engagement, education, involvement, different views. Don't presume that Indigenous communities have one view. But the most important to answer your question was the last point, which we spent quite a bit of time on, sharing the benefits of innovation with communities in an equitable manner. Arrangements may include, where appropriate, acknowledgement of contributions, return of results, local capacity building, fair or open licensing commitments, expanded access, differential pricing, and recognizing indigenous data sovereignty. So we wanted to give some examples of the range of benefits that could be part of the negotiations with indigenous communities.
Thank you. Sticking on the legal front, and I'm sure you're looking forward to splitting this particular hair. So why is it that a whole genome isn't considered personal health information in the US? And how does that work given that fine print that's expressed there? And I know you've had this conversation many times, several of us have. I'm curious to hear your response.
Yeah, this is one that we've looked at quite closely with our law firm partners and also with our HIPAA experts internally. You know, I think, fortunately for many of us companies that operate internationally, we try to kind of take adhere to the higher standard, which is generally set by the GDPR, because we have to be able to, you know, work across jurisdictions. And I would say that, you know, with respect to kind of our particular activities, for the most part, HIPAA doesn't apply to a lot of what we do. There's a limited part of our business that does do lab services. That said, it's mainly our customers' data, and we're a business associate. Sorry if eyes are starting to glaze over with that explanation, but that's, you know, so again, these are kind of, this is like a general description of I think what some of the regulatory complication is, you know, globally on this question, which is not getting any better.
Let me toss out a personal take and get anyone's reaction to this. This is an area where law and policy is behind technology and science, and that's why it's a mess. And it used to be the case that all the research useful data on a person wasn't close to being identifiable, as long as you left off their social security number, you know, and 17 other things. And it is becoming the case that the research valuable information about me includes my GPS coordinates by the second and the content of the air I breathe and everything else, and is almost impossible to de-identify, and we're in a transition state. Curious people's reactions to that, because that's why it's messy now, but where's it going?
Well, I think in one quick add-on to that is that with AI, it's only going to get worse, right? So like you can envision kind of chat GPT type, you know, scenarios where you could input a bunch of different factors about an individual and or even potentially parts of a genome sequence and potentially be able to get back to an individual a lot, you know, more easily. And so I think, you know, in the past, when you talk about, okay, well, is a whole genome sequence on its own identifiable? It's like, yes, there are videos on YouTube where you have a computer science, PhD who can show you how they can identify, an individual whole genome sequence. But that's really, it's like you have to have specialized skill and training and have like a real motivation to do it. But I think we're getting more towards the stage where it may be more accessible to re-identify a lot of different information.
Anyone else have thoughts on that before we move on?
I always think there are wiggle or wriggle, I'm not sure which verb, terms used in law when you're adjudicating these cases. And the Europeans come up with one, is there a reasonable likelihood of re-identification, so that we're not always having these conversations with these hypotheticals, what if one day somewhere, somehow someone does then, and which is very misleading to the public. Also, I think increasingly people are more at ease with sharing their genetic information, so re-identifiability isn't such a scary thing anymore. And as it becomes more normal, I mean, thanks to COVID, people talk about variants, they talk about mutations, and it isn't about Ninja Turtles. They are becoming more at home with the language of genetics and genetic variation and genetic difference and genetic uniqueness, but that it's also familial and it's also populational, subpopulational, and so on. So we're getting more genetic aware, and I think the scary part is going to disappear.
Thank you. I'm going to segue from that to the next question, which it's not directed to any of you, but I think it's good for all of you. Some of the conversations we've had within all of us around exactly this question we talk about three kinds of safety around the data. We talk about safe data, safe researchers, and safe tools. And no one of them is sufficient. The safe data is the idea of, hey, let's just restrict the data to stuff that can't possibly lead to re-identifiability. That won't work, is what we've just discussed about. But there are certainly things, including your social security number is the least safe, and then there's a spectrum. And then safe researchers is who do we give access to, and decide, and making them pinky swear that they will not do no evil. And then safe tools is can we provide environments that minimize the risk. And no one of those is sufficient, but any of the programs we work with can combine. So with that in mind, and with the safe researchers access, this question is, some industry providers can help make data sharing more scalable and sustainable. Other industries use data shared by others to develop products, drugs, and tests, not stated here, but I will add it, which then in turn can help the people who contributed the data to add complexity in both directions. Do the different cases of what kind of industry is looking for access and what the intended use is, how much does that affect the way you each think and would suggest others think about engaging with particular industry providers in the work you're doing and the programs you advise? Melissa?
So I think we look at both differently, but I think they have somewhat similar concerns around safety and in terms of the security of the data. So if the data gets into the hands of any industry and there is a breach, because I think people are more nervous about the kinds of breaches that come from industry. I think people are going to respond a lot worse if Google leaks something than if an academic institution was like, oh, I'm sorry, we had a data thing. So we are very much aware of the way that when data gets shared to industry, whether it is going to be helping to further share that data and to process it, or whether they're going to be creating a drug that's going to help the population, we see the benefit in both, but we also see that there's some caution that needs to be taken to ensure that the privacy and security standards are taken as seriously by industry as they are by governments.
Other thoughts on that?
Yeah, so we are, as you know, the pillar of the MP, the third pillar is to encourage collaborations with industry so that we can get additional funding to support other projects that are not funded by the government. So we are definitely interested to share data with industry, but we also want to be mindful of the fact that we want to protect the initial group of researchers who have contributed to the data set. So there are sort of exclusivity periods that are applied to the data set before it's open up for collaboration with industry partners and so on and so forth. At the same time, because we are forming so many different partnerships with different industry groups, we also want to be careful not to overstep certain boundaries, otherwise we run into legal issues with some of these collaborators. Yeah.
I can't remember what slide I saw it on this morning, but I know it was this morning, a concept that's been around since the Human Genome Project, and that's of pre-competitive data sharing. I think the Structural Genomics Consortium is an example of that, and how there is basic data that you don't, not everyone has to discover it on their own. There is data that is so needed by everyone, and then you can go into your separate spheres of competition. And I don't think that that concept has been developed enough, of this notion of pre-competitive data sharing amongst industry.
Thank you. I think we have time for one more question, it looks like. So let's, yeah, no, maybe not. Okay. Okay, knowing that you're keeping us from food. In Singapore, recruiting, did you have any challenges or difficulties bringing minority populations on board? And I think the word minority population has a very different resonance in Singapore than it does in some of the other countries. But how did that play out?
Yeah, so our experience is that the Chinese retired ladies, very enthusiastic. We always have oversubscription from that group of participants. Yeah, the SG100K team finds it particularly difficult to recruit the Malay and Indian ethnic group, males, 30 to 40. We don't know exactly why, but the team has been putting in a lot of effort to go into the community centers, to the mosques, as I understand, to try and encourage participation from these ethnic groups, from these minority groups.
Peter is suggesting pizza. I'm not sure that translates very well, but maybe. All right, thank you very much. Appreciate it.